There is a world on this planet where the sun has never reached. Not once. Not in all the time the oceans have existed. Below 3,000 feet, sunlight does not fade gradually. It does not dim the way a room goes dark when a lamp is switched off. It simply ends. It is absorbed, scattered, and consumed by the weight of the water above. And in that absolute blackness, something is glowing, not in a few places, not in rare pockets. Across the entire deep ocean, in every basin, in every trench, in every column of water that stretches from the twilight zone to the Hadal depths, living things are producing their own light. They flash, they pulse, they shimmer in colors that no human eye was designed to see, and the scale of this phenomenon is almost impossible to accept. Conservative scientific estimates suggest that between 80 and 90% of all creatures living below the sunlit zone are bioluminescent. That means the majority of animal life in the deep ocean, the largest habitat on the planet, generates its own light in total darkness. The deep sea is not a barren, lifeless void. It is the most widespread light show on Earth, and almost no one has ever seen it. Think about that for a moment. Humans have mapped the surface of Mars in greater detail than the floor of our own ocean. We have sent robotic rovers to photograph the dusty plains of another planet, yet we have barely glimpsed the living constellations that burn in the waters beneath our ships. The deep ocean covers more area than all the continents combined. It holds more volume than every other habitat on Earth stacked together. And in that vast space, creatures that have no access to the sun, no access to warmth, and no access to the world above, have evolved the ability to make their own light, not as a novelty, not as an accident, but as a fundamental survival strategy that touches nearly everything in the abyss. The chemistry behind it is elegant and ancient. A molecule called luciferin reacts with an enzyme called luciferase, and in the presence of oxygen, this reaction releases energy not as heat, but as photons, visible light. It is one of the most efficient chemical reactions in the known biological world. A standard incandescent light bulb loses roughly 95% of its energy as heat. Bioluminescence loses almost none. The light it produces is sometimes called cold light because it generates virtually no thermal waste. And this reaction has appeared independently in dozens of unrelated lineages across the tree of life. It is not a single trait passed down from one common ancestor. It has emerged again and again in organisms as different as bacteria and fish, as jellyfish and shrimp, as squid and worms. The deep ocean did not inherit bioluminescence. It demanded it. The pressure to survive in total darkness is so immense, so absolute, that the ability to generate light has surfaced over and over through entirely separate pathways, as though the darkness itself were issuing a command that life could not refuse. And yet, despite its extraordinary prevalence, bioluminescence remains one of the least understood phenomena in marine biology. We know it exists. We have filmed fragments of it. We have captured specimens in trawl nets and watched their light fade as they died on the deck of a ship. But we do not fully understand why so many species use it, how they coordinate it, or what messages are being exchanged in the dark. We are standing at the edge of the largest communication network on the planet, and we cannot read a single word. Every time a deep-sea camera descends into the water column, it records flashes, pulses, and glows that suggest purpose, pattern, and intent. But we lack the vocabulary to interpret them. We are watching a conversation unfold, in a language we have never studied, in a world we have never truly entered. The implications of this are staggering. If the majority of life in the deep ocean communicates through light, and if the deep ocean is the largest habitat on the planet, then the most common form of biological communication on Earth is one that humans have almost no ability to observe, record, or decode. We are not eavesdropping on an obscure dialect. We are deaf to the dominant language of the living world. And this language is not simple. It is not a single flash that means danger, or a single glow that means food. Research conducted over the past 2 decades has revealed that bioluminescent organisms produce an extraordinary range of light signals. Flashes of varying duration, pulses at different frequencies, steady glows that shift in intensity, rotating patterns that spiral across the bodies of jellyfish, color changes that move across the skin of squid faster than the human eye can follow. Some organisms produce light in 2 or 3 distinct colors, switching between them depending on context. Others produce light only from specific parts of their bodies. A chin barbel, a row of photophores along the belly, a modified fin spine, a cluster of cells behind the eye. The variety is immense, and it suggests that what is happening in the deep ocean is not a collection of isolated tricks. It is a visual ecosystem, a world in which light carries meaning, and meaning shapes survival. By the end of tonight, you are going to understand why bioluminescence is not simply a curiosity of the deep sea, but a window into a world that challenges everything we think we know about life, about light, and about the limits of biology under pressure. You are going to see how creatures that live under crushing force, in freezing water, in total darkness, have built a civilization of light that dwarfs anything on the surface. And you are going to confront a question that science has only recently begun to ask. What else is happening down there that we have never seen? Before we get started, if you find these mysteries as fascinating as we do, take a second to like the video or subscribe. It is a simple action, but it helps this channel reach more curious minds like yours. Now, let us begin. To understand why bioluminescence matters, you first have to understand the ocean itself. Not the ocean of postcards and holiday photographs. Not the turquoise shallows where children wade and surfers paddle, but the real ocean, the one that begins where the light ends and stretches downward into a darkness so complete that it has no earthly comparison. The ocean covers roughly 71% of the Earth's surface. That is a number most people have heard before, but very few have truly absorbed. It means that if you could peel the skin off the planet and lay it flat, the ocean would dominate the map so thoroughly that every continent, every mountain range, every desert, every city, and every forest would fit inside it with room to spare. The Pacific Ocean alone is larger than all the land on Earth combined. It stretches from the frozen edges of the Arctic to the coast of Antarctica, from the shores of Asia to the Americas, covering an area of approximately 63 million square miles. That is not a body of water, that is a world. And when you account for depth, the numbers become even more staggering. The average depth of the ocean is approximately 12,100 feet. That is more than 2 miles straight down. The deepest point, the Challenger Deep in the Mariana Trench, reaches nearly 36,000 feet, almost 7 miles below the surface. To put that depth in perspective, consider Mount Everest. The tallest mountain on the planet rises to a height of approximately 29,032 feet above sea level. If you could take that entire mountain, every ridge, every cliff, every frozen summit, and drop it into the Mariana Trench, its peak would still be submerged under more than a mile of water. Roughly 6,800 feet of ocean would sit above the top of Everest. The mountain would vanish. You would not see it. You would not know it was there. The water above it would continue pressing down with a force that is difficult for the human mind to process. That single comparison, the tallest structure on the surface of the Earth swallowed entirely by the depth of the ocean, should give some indication of the scale we are dealing with. But depth alone does not capture the full picture. The ocean is not just deep, it is pressurized. Pressure in the ocean increases by roughly 1 atmosphere for every 33 feet of descent. At the surface, we live under 1 atmosphere of pressure, about 14.7 pounds per square inch. That is the weight of the entire column of air above us, from the ground to the edge of space. At 1,000 feet beneath the waves, the pressure has already climbed to approximately 30 atmospheres. At 3,000 feet, where our story truly begins, it exceeds 90 atmospheres. That is the equivalent of having roughly 1,300 pounds pressing against every single square inch of your body. Imagine stacking 90 mid-sized cars on top of a single dinner plate. That is the force acting on every surface, every membrane, every cell of every organism living at that depth. And the pressure does not stop there. At 10,000 feet, it exceeds 300 atmospheres. At 20,000 feet, it passes 600. And at the bottom of the Mariana Trench, the pressure reaches over 1,090 atmospheres, more than 16,000 pounds per square inch. At that depth, the water itself becomes measurably denser. Steel structures would deform under their own weight. Glass would shatter inward in an instant. The human body, unprotected, would be compressed so quickly and so violently that consciousness would cease before the brain could register what was happening. A steel-hulled submarine designed for military operations, the kind that prowls at depths of 1,000 to 1,500 feet, would be crushed flat at less than a quarter of the way to the bottom of the Mariana Trench. The engineering tolerances required to survive at Hadal depths are so extreme that only a handful of vessels in all of human history have achieved them. The crushing weight of water at those depths is not a metaphor. It is a literal, physical reality that defines every aspect of life in the deep ocean. Yet creatures live there. They feed, they reproduce, they hunt, and they glow. They thrive under forces that would obliterate any surface organism. And their existence, more than any other single fact about the deep ocean, should tell us that the rules governing life down there are fundamentally different from the rules governing life up here. Consider another way to grasp the scale of this darkness. If you were standing at the surface of a clear, tropical ocean on a bright day, and you could watch a beam of sunlight as it penetrated downward into the water, you would see it change. In the first few feet, the light would remain strong, white, full-spectrum, energetic. But water absorbs light, and it does so selectively. Red wavelengths are absorbed first, within the top 30 or 40 feet. Orange disappears next, followed by yellow. By about 250 feet, the water has consumed everything except blue, and even the blue is fading. By 500 feet, the light has dimmed to roughly 1% of its surface intensity. By 1,000 feet, it is 1,000th of 1%, a faint, almost imperceptible trace of illumination that would be invisible to the unaided human eye. And by roughly 3,300 feet, even the most sensitive instruments cannot detect any sunlight at all. It has been absorbed completely. Every photon that entered the water from the sun above has been captured by the water molecules and converted into a tiny, unmeasurable increment of heat. The sun, for all its power, cannot reach the deep ocean. Its light dies in the water column, and below that point there is no external source of illumination, none. The only light that exists in the midnight zone, the abyss, and the Hadal trenches is light made by living things. To put the timescale of this darkness in perspective, consider that the speed of light is approximately 186,000 miles per second. A photon leaving the surface of the sun takes roughly 8 minutes and 20 seconds to reach the Earth. That same photon, upon striking the surface of the ocean, would penetrate downward through the water column and be fully absorbed. Extinguished within a fraction of a second, it travelled 93 million miles through the vacuum of space in about 8 minutes, and then it died in the first few hundred feet of ocean water. The deep ocean is so effective at blocking light that even the most powerful natural light source in the solar system cannot push a single photon past the twilight zone. The darkness below is not the absence of a weak light source. It is the defeat of the strongest one. To understand the world these creatures inhabit, it helps to know how the ocean is divided. Marine scientists have organized the water column into 5 major zones, each defined by depth, light, temperature, and pressure. These are not arbitrary categories. They represent fundamentally different environments, each with its own physical rules and biological inhabitants. Knowing these zones is essential, because bioluminescence does not appear uniformly throughout the ocean. It follows a pattern, and that pattern maps directly onto the structure of the water column. The first zone is the epipelagic, sometimes called the sunlight zone. It extends from the surface to approximately 660 feet. This is where the vast majority of marine life that humans are familiar with exists. Coral reefs, tuna, dolphins, sea turtles, whale sharks, and the microscopic phytoplankton that produce roughly half of the oxygen on the planet. Sunlight penetrates this zone with enough intensity to fuel photosynthesis, the process by which plants and algae convert light into chemical energy. The epipelagic is warm, comparatively bright, and teeming with life. It is the ocean as most people imagine it, colourful, dynamic, and populated by species that appear on nature programs and in aquarium exhibits. It is also the thinnest of the 5 zones, a narrow band of illuminated water sitting on top of a vastly larger body of darkness. Below the sunlight zone lies the mesopelagic zone, also known as the twilight zone. This region spans from approximately 660 feet to roughly 3,300 feet. Here, sunlight has not vanished entirely, but it has weakened to a ghostly residue, a pale, directionless shimmer that is enough for some organisms to detect, but nowhere near sufficient for photosynthesis. The light that remains is almost entirely blue because water absorbs red, orange, yellow, and green wavelengths at shallower depths. By the time sunlight reaches the lower mesopelagic, it has been filtered into a dim, monochromatic glow that fades imperceptibly into nothing. The temperature in the twilight zone drops sharply, the pressure climbs, and something profound begins to happen. In this half-lit world, bioluminescence starts to appear, not as a rarity, but as a defining characteristic of the environment. Animals in the mesopelagic use their own light to hunt, to hide, to communicate, and to deceive. The twilight zone is a transition, a border between the world of the sun and the world of self-made light. It is home to an extraordinary diversity of organisms, many of which participate in the daily vertical migration, rising toward the surface at night to feed and descending back into the twilight at dawn. This migration involves billions of organisms and moves more biomass than any other animal movement on the planet, and it is accompanied, at every stage, by bioluminescent activity. Then comes the bathypelagic zone, the midnight zone, stretching from approximately 3,300 feet down to around 13,000 feet. Here, the sun is gone. Completely. Not a single photon of sunlight reaches this depth. The darkness is absolute, and it is permanent. No sunrise will ever reach the midnight zone. No season will brighten it. No weather system, no volcanic eruption, no event on the surface of the planet will ever send a ray of natural light into this space. The temperature hovers just above freezing, between roughly 2 and 4 degrees Celsius in most ocean basins. The pressure is immense. Food is scarce, arriving only as a slow, continuous rain of dead organic material from the waters above, a phenomenon scientists call marine snow. And yet, the midnight zone contains one of the largest ecosystems on the planet by sheer volume. It is home to an astonishing array of life. Anglerfish with their dangling lures, giant squid that eluded direct observation in their natural habitat until the early 2000s, siphonophores that stretch longer than blue whales, and countless species that remain entirely unknown to science. The midnight zone is where bioluminescence becomes not just common but dominant. Here, the overwhelming majority of organisms can produce light. In this realm, bioluminescence is the rule, not the exception. It is the primary visual experience of every creature that lives here. Below the midnight zone lies the abyssopelagic zone, commonly called the abyssal zone. This extends from approximately 13,000 feet to roughly 20,000 feet. The word abyss comes from the Greek word abyssos, meaning bottomless, and it is a fitting name. The abyssal plains are vast, flat expanses of sediment that cover more of the Earth's surface than any other landscape on the planet. They are the true floor of the ocean, enormous, flat deserts of fine silt and clay, interrupted only by occasional seamounts, ridges, and the remains of ancient volcanic activity. These plains are covered in a slow, unending accumulation of marine snow, the compressed remains of organisms that lived and died in the waters above, drifting downward over days, weeks, and months before finally settling on the bottom. The temperature is near freezing. The pressure exceeds 400 atmospheres. Life here is sparse but present, adapted to conditions that would kill most surface organisms in seconds. Brittle stars crawl across the sediment. Sea cucumbers vacuum the organic particles from the mud. Ghostly, translucent fish drift through the water just above the bottom, their bodies so fragile that the slightest touch could rupture their tissues. Finally, below the abyss, in the deepest ocean trenches, lies the Hadal Zone, named after Hades, the Greek underworld. This zone begins at approximately 20,000 feet and extends to the very bottom of the ocean, the Challenger Deep, at nearly 36,000 feet. Only a handful of trenches on Earth reach Hadal depths: the Mariana Trench, the Tonga Trench, the Kuril-Kamchatka Trench, the Philippine Trench, the Kermadec Trench. These are not smooth slopes descending gently into darkness. They are narrow, steep-sided fractures in the ocean floor, created where tectonic plates collide and one plate is forced beneath another in a process called subduction. The organisms that live in these trenches are among the most isolated on the planet. Some species found in one Haddle Trench have been found in no other location on Earth, separated by thousands of miles of deep water that they cannot cross. They are prisoners of geography, confined to individual canyons at the bottom of the sea. Now, here is the critical detail. The sunlit zone, the epipelagic, represents less than 5% of the ocean's total volume. Everything below it, the twilight zone, the midnight zone, the abyss, and the Haddle Trenches, makes up the remaining 95% or more. That means the vast majority of the ocean exists in darkness. The ocean that humans see, the ocean of beaches and blue horizons, is a thin, sunlit skin stretched over a body of darkness that is almost incomprehensibly large. If the ocean were a building, the sunlit zone would be the paint on the roof. Everything below it, floor after floor, basement after basement, descending into the earth, would be the building itself. And in that darkness, the dominant form of visible light is not the sun. It is biology. To fully appreciate what this means, consider the contrast between the surface and the deep in terms of food. The sunlit zone produces virtually all of the ocean's organic matter through photosynthesis. Phytoplankton, microscopic algae that float in the upper water column, capture sunlight and use it to build organic molecules from carbon dioxide and water. These tiny organisms are consumed by zooplankton, which are consumed by small fish, which are consumed by larger fish, and so on up the food chain. The sunlit zone is a factory of biological production, and it produces more organic carbon per year than all the forests on land combined. But this productivity is confined to the top 200 feet of the ocean. Below that, production drops to zero. The deep ocean produces almost no food of its own. Instead, the deep relies on what falls from above. Marine snow, the constant, slow rain of dead phytoplankton, fecal pellets, shed exoskeletons, decomposing jellyfish, and other organic debris, drifts downward through the water column at rates of roughly 100 to 200 meters per day. A particle of marine snow leaving the sunlit zone might take weeks to reach the abyssal floor, and during its descent, much of it is consumed, decomposed, or dissolved by the organisms it passes through. By the time marine snow reaches 4,000 feet, roughly 90% of the original organic matter has been removed by organisms in the mesopelagic and upper bathypelagic. By the time it reaches the abyssal floor at 13,000 to 20,000 feet, the remaining material is a tiny fraction of what was produced at the surface, typically less than 5%, and often less than 1%. This means that the deep ocean is, from an energy perspective, a desert. The organisms living there subsist on the crumbs that fall from a table miles above. Every calorie is precious. Every metabolic expenditure must justify itself. Waste is not an option. Inefficiency is punished by starvation. And yet, in this environment of extreme scarcity, the overwhelming majority of organisms devote metabolic resources to producing light. They synthesize luciferin. They build photophores. They maintain the enzymatic machinery necessary for bioluminescence. They flash, they glow, they pulse, spending energy that could otherwise be used for growth, reproduction, or movement. The fact that bioluminescence persists under these conditions, that it has not been eliminated by the ruthless economics of deep-sea survival, is perhaps the strongest possible argument for its importance. In an environment where every calorie counts, you do not waste energy on something trivial. If nearly every organism in the deep ocean produces light, it is because light is essential. The history of our understanding of this realm is surprisingly short. For most of human civilization, the deep ocean was assumed to be empty. It was regarded as a wasteland, a cold and lifeless desert beneath the productive surface waters. In the mid-1800s, a prominent British naturalist named Edward Forbes proposed what became known as the azoic hypothesis, the idea that no life could exist below approximately 1,800 feet. Forbes based this on his own dredging expeditions in the Aegean Sea, where he found that the number of organisms decreased with increasing depth. He drew a line on the graph, extended it downward, and concluded that by about 300 fathoms, life would reach zero. The deep ocean floor, in Forbes's view, was a lifeless desert of mud and darkness. This was not a fringe idea. It was accepted by much of the scientific community for nearly 2 decades. It appeared in textbooks. It was cited in lectures. It shaped the way an entire generation of naturalists thought about the ocean. It was wrong. The first serious challenge to the azoic hypothesis came in 1860, when a damaged telegraph cable was hauled up from a depth of approximately 6,000 feet in the Mediterranean Sea. The cable was encrusted with living organisms, corals, mollusks, worms, and other invertebrates that had colonized it in the dark. If life existed at 6,000 feet, the azoic hypothesis was finished. The telegraph cable incident was remarkable not only for what it proved, but for how it proved it. The cable had been laid by engineers who had no interest in biology. It had sat on the ocean floor for years, accumulating life without any scientific intention or oversight. The organisms that encrusted it were not specimens collected by a carefully designed expedition. They were accidental passengers, hauled to the surface as an unintended consequence of industrial maintenance. The discovery was a gift from the telecommunications industry to the biological sciences. An accidental experiment conducted at a depth no biologist of the era could reach, over a time frame no laboratory could replicate, under conditions no one had thought to test, and its message was unmistakable. The deep ocean floor was not a barren wasteland. It was a substrate, a surface, a habitat, a place where organisms could settle, attach, grow, and reproduce in the permanent darkness, under hundreds of atmospheres of pressure, miles from the nearest photon of sunlight. Other incidental evidence accumulated over the following decade. Sounding lines hauled up from great depths sometimes returned with organisms tangled in their fibers. Deep-water fishing operations in the North Atlantic occasionally brought up species that no one could identify, pale and strange and clearly adapted to conditions that no surface organism could tolerate. Norwegian naturalist Michael Sars and his son, Georg Ossian Sars, collected numerous deep-water invertebrates from the fjords of Norway, documenting species at depths well below Forbes's supposed limit of life. Each discovery chipped away at the azoic hypothesis, but none delivered the decisive blow. That required something more ambitious. But the full demolition came with the voyage of HMS Challenger, a British Royal Navy corvette that set sail in 1872 on what would become the most important oceanographic expedition in history. Under the scientific direction of Charles Wyville Thomson, and later analysed by John Murray, the Challenger expedition covered nearly 70,000 nautical miles across the Atlantic, Pacific, Indian, and Southern Oceans. Over the course of nearly 4 years, the crew took hundreds of deep-sea soundings, measured temperatures at depth, collected water samples, and hauled up biological specimens from depths that had never been sampled before. The expedition discovered over 4,700 new species. It proved beyond any doubt that life existed at all depths of the ocean, including the very bottom. It found organisms in the deepest soundings, in the coldest water, and in the most remote basins. The azoic hypothesis was not merely disproven, it was obliterated. Forbes's neat, downward trending line was replaced by a far more complex and far more humbling reality. Life did not decrease uniformly with depth. It changed form. It changed density. It adapted in ways that no surface biologist had predicted. But it did not disappear. Not at 1,000 feet. Not at 10,000 feet. Not at 20,000 feet. Among the many discoveries made by the Challenger expedition was the first measurement of the Mariana Trench. In March of 1875, the crew took a sounding in the western Pacific that recorded a depth of approximately 26,850 feet, a number that astonished the scientific world. Later measurements with more precise instruments would reveal the trench to be even deeper, but the Challenger's initial sounding established the existence of ocean depths that were far beyond anything previously imagined. And yet, even after the Challenger expedition, the study of deep-sea life remained painfully slow. The technology simply did not exist to observe these organisms in their natural environment. You cannot put a microscope at the bottom of the ocean. You cannot keep a deep-sea fish alive in a surface laboratory for extended study. Every specimen brought to the surface underwent massive decompression, dramatic temperature shock, and sudden light exposure. By the time scientists could examine them, these creatures were often damaged, deformed, discolored, or dead. Their bioluminescence, perhaps their most remarkable and definitive feature, was almost always gone. The very trait that made them extraordinary in life was the first thing destroyed by the process of collection. The first manned descent to truly extreme depths came in 1960 when Swiss oceanographer Jacques Piccard and United States Navy Lieutenant Don Walsh descended in the bathyscaphe Trieste to the bottom of the Challenger Deep. They reached a depth of approximately 35,814 feet. The descent took nearly 5 hours. The cabin was a steel sphere just 7 feet in diameter, barely large enough for 2 men to sit side by side. The walls of the sphere were 5 inches thick, forged from specially treated steel designed to withstand the crushing pressure of the deepest ocean. As they descended, the men could hear the metal creaking and groaning under the increasing load. Sounds that reminded them, with every passing minute, that the margin between survival and catastrophic implosion was measured in fractions of an inch of engineered metal. At the bottom, they spent roughly 20 minutes peering through a single small porthole into a cloud of sediment stirred up by their landing. They reported seeing what appeared to be a flatfish on the bottom, though this observation was later debated by marine biologists. What they could not see, because their lights illuminated only a tiny circle in a vast field of darkness, and because the sediment cloud obscured everything beyond a few feet, was the bioluminescent world that surrounded them. They had reached the deepest point on Earth, and they had seen almost nothing. It was not until the development of modern deep-sea submersibles, remotely operated vehicles, and high-definition cameras in the late 20th century that scientists began to grasp the true extent of bioluminescence in the deep ocean, and what they found was astonishing. In expedition after expedition, researchers reported that the majority of organisms they encountered in the deep were bioluminescent. Not a minority, not a specialist group, the majority. The deep ocean was not dark in the way they had assumed. It was filled with light, biological light, generated by the organisms themselves, pulsing and flashing in patterns that suggested communication, defense, predation, and behaviors that scientists could not yet interpret. The first large-scale systematic survey of deep-sea bioluminescence was conducted in the early 2000s, when researchers from the Monterey Bay Aquarium Research Institute, commonly known as MBARI, analyzed thousands of hours of remotely operated vehicle footage from Monterey Canyon off the coast of California. The results, published by marine biologist Stephen Haddock and his colleagues, confirmed what many deep-sea explorers had suspected. In the mesopelagic and bathypelagic zones, bioluminescent organisms accounted for approximately 76% of all observed individuals. In some regions and depth ranges, that number exceeded 90%, 76%. That is not a minor fraction, that is the overwhelming majority. It means that if you could somehow transport yourself to a depth of 3,000 feet in the open ocean and look around with eyes adapted to the dark, nearly 8 out of every 10 living things you would encounter would be capable of producing their own light. The deep sea is not a place where bioluminescence occasionally appears. It is a place where bioluminescence is the norm, the baseline, the background condition against which all other adaptations are measured. It is worth pausing to consider what the MBARI data actually represents and what its limitations are. The 76% figure comes from video footage captured by remotely operated vehicles operating with bright artificial lights, the very instruments that, as we have discussed, alter the behavior of deep-sea organisms and potentially drive the most light-sensitive species away from the camera. If the organisms that avoid brightly lit vehicles are disproportionately bioluminescent, and there is every reason to believe that they are, since bioluminescent organisms tend to have the most sensitive eyes, then the 76% figure is an undercount. The true proportion of bioluminescent organisms in the deep ocean may be 80% or 90% or higher. The MBARI study was groundbreaking, but it was conducted with tools that systematically underrepresent the very phenomenon it was trying to measure. That realization alone should unsettle any assumption we hold about how well we understand the deep. And this raises a question that is far more unsettling than it first appears. If the deep ocean, the largest living space on the planet, is dominated by organisms that produce light, then why do we know so little about it? Why has this phenomenon, which is arguably more widespread than photosynthesis in terms of the volume of habitat it dominates, received so little sustained scientific attention? The answer has to do with the sheer difficulty of observing it, and with a set of biases so deep in our thinking that we rarely examine them. Every tool we use to study the deep sea is in some fundamental way an intrusion. Submersibles bring bright artificial lights that overwhelm the delicate flashes of bioluminescence. Trawl nets drag organisms to the surface, killing them and destroying the light displays they would have produced in life. Remotely operated vehicles generate noise, vibration, and electromagnetic fields that can alter the behavior of the creatures they are meant to observe. We have been trying to study a light show by flooding the stage with spotlights. We have been trying to listen to a whisper by shouting into the darkness. And the result is that our understanding of deep-sea bioluminescence remains, even now, profoundly incomplete. But what we do know is extraordinary, and what scientists have begun to discover in recent years is forcing a fundamental reconsideration of what the deep ocean is, how it works, and what it means for our understanding of life on this planet. What follows is the story of that discovery, and the questions it has raised that no one can yet answer. The central mystery of deep-sea bioluminescence is not that it exists. It is that it exists so abundantly, in so many forms, across so many unrelated species, and with such apparent complexity that no single explanation can account for it. It is as though the deep ocean has developed its own language, a language written in light, and we have only just begun to recognize the alphabet. To understand the scope of the problem, consider how many different ways bioluminescence is used beneath 3,000 feet. This is not a single trait with a single function. It is a suite of behaviors, mechanisms, and strategies that are as varied as the organisms that employ them. Each use tells a different story about survival in the dark, and each one raises questions that the others do not answer. The most famous example is the anglerfish. The deep-sea anglerfish, of which there are more than 200 known species, carries a bioluminescent lure called an esker on the end of a modified dorsal spine that extends from its head like a fishing rod. The light in this lure is not produced by the fish itself. It is produced by symbiotic bacteria, bioluminescent microorganisms that live inside the esker and generate light continuously or in response to chemical signals from the host. The anglerfish dangles this lure in the darkness, and prey, small fish, shrimp, and other organisms attracted to the light, swim toward it. When they get close enough, the anglerfish strikes. Its jaws can open wide enough to consume prey nearly twice its own body size, a necessity in an environment where meals are rare and unpredictable. The entire predatory strategy depends on light. Without bioluminescence, the anglerfish would be sitting blind and hungry in an empty void, waiting for food to stumble into its mouth by pure chance. The anglerfish's dependence on its lure is so total that the entire body plan of the animal is organized around it. The fish does not actively swim in search of prey. It hangs motionless in the water column, expending as little energy as possible, dangling its esker and waiting. Its body is designed for ambush, a large mouth, expandable stomach, and minimal musculature for sustained swimming. It is a mouth with a fishing rod attached, and the fishing rod works only because of the bacteria that live inside it, producing a glow that is calibrated to attract the specific organisms that the anglerfish eats. Some species of anglerfish produce a steady, dim glow. Others produce rhythmic pulses of light. Still others can modulate the intensity and color of their lure, brightening it to attract attention and dimming it when prey is close, to avoid startling the target at the last moment. The sophistication of this lure behavior suggests a level of control over the bacterial light source that is not well understood. The fish and the bacteria appear to be in communication, a biochemical conversation in which the fish regulates the light output of its symbiont, and the symbiont responds. But the anglerfish also reveals a deeper puzzle. The relationship between the fish and its bacteria is obligate. Neither can survive well without the other. The bacteria require the sheltered, nutrient-rich environment of the esker. The fish requires the light. But how did this partnership begin? How did a bacterium come to live inside a specialized organ on a fish's head, producing light on demand? The answer involves a degree of codependence that is difficult to explain through any simple mechanism. The bacteria must have colonized the esker, or an ancestral version of it, at some point in the past, but the esker itself is a highly specialized structure that seems to exist specifically to house bacteria. Which came first, the organ that houses the bacteria, or the bacteria that made the organ useful? This is a version of the chicken and egg problem, and in the deep ocean, it plays out in dozens of different species, each with its own variation on the theme. The complexity of the anglerfish-bacterium partnership extends further than most accounts acknowledge. The bacteria inside the esker are not simply passive tenants producing light as a byproduct of their metabolism. They are maintained, regulated, and in some respects imprisoned by their host. The esker contains a network of fine tubules through which the host fish can control the flow of oxygen to the bacteria. When oxygen is abundant, the bacteria glow brightly. When the fish restricts oxygen, the light dims or goes out entirely. The fish can also release chemical signals, hormone-like molecules, that modulate bacterial gene expression, effectively turning specific bacterial functions on or off. This is not a loose association between two organisms that happen to share a space. It is an intimate, bidirectional biochemical relationship in which the host controls the symbiont with a level of precision that borders on the physiological, and the bacteria, for their part, appear to have lost the ability to survive independently. Anglerfish esker bacteria, when cultured outside the host in laboratory conditions, often fail to grow or lose their bioluminescence within a few generations. Their genomes have contracted over time, shedding genes that would be necessary for free-living survival. They have become obligate symbionts, organisms so deeply enmeshed in the partnership that they can no longer function alone. The relationship has passed the point of no return. The fish cannot hunt without the bacteria. The bacteria cannot survive without the fish. They are locked together in a dependence so complete that they might as well be considered a single organism, a chimera, part animal and part bacterium, existing in the darkness and hunting by a light that neither partner could produce alone. What makes this even more remarkable is that the anglerfish is not a single case. There are more than 200 species of deep-sea anglerfish spread across multiple families that diverged from each other millions of years ago. Each species has its own variation of the esker, its own bacterial symbiont or combination of symbionts, its own flash pattern, its own lure behavior. The anglerfish model, predation by symbiotic bioluminescent lure, has been reinvented, refined and diversified across hundreds of independent lineages, each adapting the basic concept to its specific environment, prey type, and depth range. The diversity within this single strategy alone is staggering, and the anglerfish is only one of dozens of fundamentally different approaches to bioluminescence in the deep. Consider the firefly squid, Wattersonia scintillans, found in the deep waters off Japan. This small squid, typically no more than 3 inches long, possesses hundreds of photophores, specialized light-producing organs, embedded in its skin, its tentacles, and around its eyes. Unlike the anglerfish, the firefly squid does not rely on bacteria. It produces its own light using its own chemistry, and it can produce that light in 3 different colors: blue, green, and a rare deep red. It uses this light for counter-illumination, a technique in which the squid matches the faint, downwelling light from above by producing its own light on its underside, effectively erasing its own silhouette when viewed from below. From beneath, a predator looking upward would see no dark shape against the faint glow of the twilight zone. The squid becomes invisible, merged with the ambient light field. This is not a simple on-off switch. The firefly squid adjusts the intensity, color, and pattern of its bioluminescence in real time, responding to changes in the ambient light conditions as it moves through different depths and as the overhead light shifts with the time of day and the clarity of the water. It is dynamic camouflage, executed in 3 dimensions, using a chemistry that humans can barely replicate in a laboratory. The precision required is extraordinary. The squid must detect the light coming from above, calculate the appropriate counter-illumination pattern for its own size and orientation, generate that pattern across hundreds of individually controlled photophores, and maintain the match as conditions change. All of this happens automatically, continuously, and without conscious thought, in an animal whose brain is smaller than a grain of rice. Counter-illumination is not unique to the firefly squid. It is one of the most widespread uses of bioluminescence in the mesopelagic zone, employed by dozens of species of squid, fish, shrimp, and other organisms. The hatchetfish, a small deep-sea fish with a compressed blade-like body, uses rows of photophores along its ventral surface to match the downwelling light with extraordinary precision. Researchers who have measured the light output of living hatchetfish have found that the photophores can adjust their brightness in response to changes in ambient light within fractions of a second. The match is so accurate that when viewed from below, the fish effectively disappears, its dark silhouette erased by the glow from its own belly. The hatchetfish becomes a hole in the water, indistinguishable from the faint light field above. Predators looking upward see nothing. The camouflage is virtually perfect. The lanternfish, the most abundant group of vertebrates on the planet by some estimates, also uses counter-illumination extensively. Lanternfish, small, schooling fish that dominate the mesopelagic zone in every ocean, possess dense arrays of photophores arranged in species-specific patterns along their bodies. These patterns serve a dual purpose: counter-illumination during the day, when the fish hang at depth and risk being spotted from below, and species recognition during the vertical migration at night, when vast mixed schools of lanternfish ascend toward the surface to feed. The photophore patterns are as distinctive as fingerprints, allowing individuals to identify members of their own species in the dark, to form cohesive schools, and to find mates among thousands of similar-looking fish. Then there are the organisms that use bioluminescence for defense in ways that are startling in their ingenuity. The deep-sea shrimp Acanthophera purpurea, when attacked by a predator, does not simply flee. It vomits a cloud of bioluminescent fluid directly into the face of its attacker. The sudden burst of light is blinding, the deep-sea equivalent of a flashbang grenade thrown in a dark room. In an environment where eyes are adapted to detect the faintest glimmers, a sudden explosion of bright blue light is overwhelming. While the predator recoils, momentarily stunned and temporarily unable to see, the shrimp jets backward into the darkness and escapes. The mechanics of this defense are worth examining in detail because they reveal the sophistication of the underlying biology. The shrimp stores bioluminescent material in a specialized gland near its mouth. The material is a complex mixture containing luciferin, luciferase, and various binding proteins that keep the reaction from occurring prematurely. When the shrimp detects the contact of a predator through mechanoreceptors on its antennae and body surface, it triggers a rapid muscular contraction that expels the material through its mouth in a directed jet. The timing, the volume, and the direction of the discharge are all controlled, and the reaction begins immediately upon contact with the surrounding seawater, producing an intense blue flash that can last for several seconds. The entire sequence, detection, decision, expulsion, and escape, occurs in a fraction of a second. It is a reflex honed to near perfection by the relentless pressure of predation in an environment where there are no hiding places, no cover, no terrain to use as shelter. In the open water column of the midnight zone, you cannot hide behind a rock or dive into a crevice. You can only blind your attacker and run. The metabolic cost of this defense is significant. The bioluminescent material must be continuously manufactured and stored, consuming resources that could otherwise be devoted to growth or reproduction. Once expended, it must be regenerated, which takes time during which the shrimp is vulnerable to a second attack. Some researchers have estimated that a shrimp may be able to deploy its bioluminescent defense only a limited number of times before exhausting its reserves, and that recovery between deployments may take hours or even days. This creates a tactical dilemma for the shrimp that parallels problems in military strategy. When do you use your most powerful weapon? Too early, and you waste resources on a false alarm. Too late, and you are already dead. The shrimp must assess the threat, decide whether to deploy, and execute the defense at precisely the right moment, with no room for error and no opportunity for a second attempt if the first one fails. Other species have developed equally creative defensive strategies. Certain brittle stars, when seized by a predator, can detach their own arms at predetermined fracture points. The severed arm continues to glow and twitch after separation, writhing on the sea floor or drifting in the water, creating a decoy that attracts the predator's attention while the main body of the brittle star crawls away into the darkness to regenerate. Some species of sea cucumbers take this even further. When threatened, they can expel their own internal organs, their intestines and respiratory structures, in a mass of sticky, bioluminescent tissue that entangles and confuses the attacker. The expelled organs glow in the water, pulsing with light, while the sea cucumber retreats. It will regrow its internal organs over the following weeks, a regenerative ability that seems almost impossible by the standards of surface biology. But in the deep ocean, where escape options are limited and encounters with predators can be lethal, the ability to sacrifice a glowing piece of yourself to buy time for escape is a survival strategy of immense value. Some organisms use bioluminescence not to hide or to escape, but to call for help. This is known as the burglar alarm hypothesis, and it is one of the most elegant ideas in deep-sea biology. The concept is simple, but its implications are profound. When a small bioluminescent organism is seized by a mid-sized predator, it produces a brilliant flash, or a series of rapid, intense flashes. These flashes are not aimed at the predator that is currently eating it. They are aimed outward, into the darkness, at an even larger predator. The light display is a distress signal, a beacon that says, in effect, something is feeding here. The flashes attract the attention of a bigger animal which is drawn to investigate. If the bigger predator attacks the mid-sized predator, the smaller organism may be released or forgotten in the chaos of the encounter. In effect, the prey is calling a bigger bully to come and save it. The deep sea is so dark that any light signal can be seen from a considerable distance, making this strategy far more effective than it would be in a well-lit environment where visual signals compete with ambient light. In the pitch blackness of the midnight zone, a single bright flash can travel hundreds of feet through the clear, particle-free water, reaching the eyes of predators that the original attacker could never detect. The burglar alarm is a survival strategy that exploits the very darkness that creates the need for bioluminescence in the first place. The elegance of the burglar alarm hypothesis is matched by its disturbing implications. If small organisms routinely attract large predators by lighting up during an attack, then the entire predatory food web of the deep ocean is connected by bioluminescent signals. A shrimp attacked by a small fish produces light that attracts a larger fish, which may in turn be attacked by a squid that produces its own light, attracting an even larger predator, and so on up the chain. Each predation event generates a light signal. Each signal draws attention. Each response generates new signals. The result is a cascading network of bioluminescent events that ripple outward from any initial predator-prey encounter, propagating through the water column like a chain reaction. A single attack in the midnight zone does not end when the prey is consumed. It echoes outward in light, attracting attention, provoking responses, and reshaping the behavior of organisms across a wide area. This cascade effect means that the darkness of the deep ocean is never truly still. At any given moment, in any given cubic mile of deep water, something is being attacked, something is flashing, something is responding. The midnight zone may appear empty and featureless to a human descending in a submersible, but the apparent emptiness is an illusion created by the scale of the environment and the limitations of the observer. If we could see all the bioluminescent signals in a single cubic mile of deep water simultaneously, every flash, every glow, every pulse, we would see a landscape of constant flickering activity, a visual field as dynamic and complex as the canopy of a rainforest or the streets of a city at night. We cannot see it, but it is happening constantly, everywhere, in the dark, and it gets worse, or rather, it gets stranger, because beyond these individual examples lies a pattern that no one fully understands, a pattern of diversity and complexity that exceeds what any simple functional explanation can account for. In 2009, a team of researchers led by marine biologist Edith Widder deployed a device called the Eye in the Sea, an unlit, remotely operated camera system designed to observe deep-sea life without disturbing it. Unlike traditional submersibles and remotely operated vehicles, the eye in the sea used no white light. Instead, it relied on red illumination, which most deep-sea organisms cannot detect, and a bioluminescent lure called an electronic jellyfish, a ring of blue light-emitting diodes designed to mimic the flash pattern of a common deep-sea jellyfish, Atolla weevillei. The camera sat motionless on the seafloor, dark and silent, waiting. The results were transformative. Within minutes of activating the electronic jellyfish lure, Widder's camera recorded organisms that had never been seen before. Not rare species that had occasionally appeared in trawl catches or on other camera deployments. Species that were entirely unknown to science. Creatures that had never appeared in any trawl net, never been filmed by any submersible, and never been described in any scientific paper. They emerged from the darkness, drawn by the light, and they were recorded by a camera that did not scare them away. This single experiment revealed something that had been suspected but never proven with such clarity. The deep ocean contains a vast population of organisms that are not merely difficult to observe. They are specifically adapted to avoid detection by the tools we use. They flee from the noise and vibration of submersibles. They are killed by the pressure changes in trawl nets or damaged beyond recognition during the haul to the surface. They are blinded and startled by the intense white lights of remotely operated vehicles, retreating long before the camera lens can focus on them. The organisms that we have catalogued from the deep sea are not a representative sample of what lives there. They are the fraction that happen to be slow enough, tough enough, or unlucky enough to be captured by our clumsy instruments. The scope of this observational failure is difficult to overstate. Consider what it means for every prior estimate of deep-sea biodiversity, every population count, every species census, every ecological model built on data collected by trawl nets, submersibles, and brightly lit cameras. All of those estimates are underestimates. All of those models are incomplete. Every number we have cited about how many species live in the deep, how many are bioluminescent, how dense the populations are, all of those numbers must be treated as lower bounds, not true values. The real numbers are higher. How much higher is the question that haunts deep-sea biology, and the answer, based on the Widder experiments and others like them, is much higher than we have assumed. The eye in the sea did not merely discover new species. It demonstrated that the observational gap between what we have seen and what exists is not a narrow margin of error. It is a gulf, a structural, systematic, pervasive gap in our knowledge that affects every conclusion we have drawn about the deep ocean. The new species that appeared before Widder's camera were not hiding in an unusual corner of the ocean. They were in an ordinary patch of deep water, at an ordinary depth, in an ordinary region. They were everywhere. They had always been everywhere. We had simply been looking the wrong way. Widder herself has described our current approach to deep-sea observation as the equivalent of trying to survey the wildlife of the African savanna by driving through it at night in a dump truck with the headlights on and the horn blaring. Whatever you see under those conditions, a startled gazelle frozen in the headlights, a frightened bird taking flight, would not be a reliable census of what actually lives there. You would miss the lions hiding in the grass. You would miss the nocturnal predators that fled before you arrived. You would conclude that the savanna was populated almost entirely by a few species of confused, slow-moving herbivores. And you would be wrong. The same, Widder argues, is true of the deep ocean. And bioluminescence, the very trait that defines the majority of life in the deep, is the trait most easily destroyed and most thoroughly disrupted by our methods of observation. But here is where the mystery deepens into something that approaches the genuinely unsettling. If bioluminescence is so common, so widespread, and so fundamental to deep-sea survival, then how do we explain the sheer diversity of its forms? Why does one species produce blue light while another produces green? Why do some organisms flash in rapid bursts while others glow with a steady, continuous radiance? Why do some use bacteria as their light source while others synthesize their own luminescent chemistry from scratch? And why has bioluminescence arisen independently at least 50 separate times across the tree of life, through entirely separate biochemical pathways? The last question is particularly revealing, and it deserves extended attention because it touches on one of the most fundamental questions in biology. What makes certain traits appear again and again, independently, in organisms that share no recent common ancestor? This pattern, called convergent development, occurs when different lineages face the same environmental pressures and arrive at the same solution through entirely different genetic and biochemical routes. Wings developed independently in birds, bats, and insects. Eyes developed independently in vertebrates and cephalopods. Streamlined body shapes developed independently in dolphins, sharks, and ichthyosaurs. Each case of convergent development tells us that the solution is not a random accident. It is an optimal response to a specific environmental challenge, a response so effective that life finds it again and again, no matter what starting materials are available. Bioluminescence is one of the most dramatic examples of convergent development in the history of life. At least 50 independent origins, using different chemistries, different enzymes, different organ structures, in organisms as different from each other as a bacterium and a shark. The sheer number of independent origins tells us something important. Whatever bioluminescence does for organisms in the deep ocean, it does it so well that the pressure to develop it is essentially irresistible. Any lineage that enters the deep ocean and remains there long enough will eventually develop bioluminescence or acquire it through symbiosis, because the organisms that have it survive and reproduce at dramatically higher rates than those that do not. The deep ocean does not merely permit bioluminescence, it demands it. On land, bioluminescence is relatively rare. Fireflies are the most familiar example, along with certain species of fungi, glowworms, and a few beetles. But in the ocean, bioluminescence is everywhere. It occurs in bacteria, in single-celled dinoflagellates that light up tropical bays with ghostly blue fire, in jellyfish, in comb jellies, in polyquite worms, in crustaceans, in squid, in octopuses, in fish, and even in certain species of sharks. It occurs at every depth, in every ocean basin, and in both the water column and on the seafloor. The contrast between land and sea is so stark that it demands an explanation. The most widely accepted explanation is environmental. In the deep ocean, where there is no sunlight, bioluminescence is the only form of visual communication available. It is not a luxury, it is a necessity. Any organism that can produce light in total darkness has a significant advantage in finding mates across vast expanses of empty water, in attracting prey that would otherwise pass invisibly in the dark, in escaping predators through confusion and misdirection, in signaling distress, in marking territory. Light, in the absence of the sun, becomes the most valuable currency in the ecosystem. The pressure to evolve bioluminescence is so intense that it has driven organism after organism, lineage after lineage, to independently develop the same capability. But the problem is that this broad explanation, while correct in its general outline, does not account for the specifics. It does not explain why the dragonfish Malacosteus niger has evolved the ability to produce far-red bioluminescence. Light at a wavelength of approximately 700 nanometers, which is nearly invisible to the vast majority of deep-sea organisms. Most deep-sea eyes are tuned to blue light, at wavelengths around 470 to 490 nanometers. Blue is the color that travels farthest through seawater. Blue is the color that most bioluminescent organisms produce. Blue is the color that deep-sea eyes have been optimized to detect, and Malacosteus has developed a spotlight that operates entirely outside that range. It sees in red. Its prey sees only in blue. The dragonfish can illuminate its targets without their knowledge. It has essentially developed its own private channel of light, a sniper's infrared scope in a world of blue-blind victims. This is not a simple adaptation. It requires specialized photophores that produce near-infrared photons, and it requires eyes with unique pigments, specifically a chlorophyll-derived photosensitizer that can detect those photons. Both traits must exist together for the strategy to work. Each alone is entirely useless. The Malacosteus case illustrates a broader principle that applies throughout the bioluminescent deep ocean. The organisms are not merely producing light, they are engaged in an arms race. For every defensive use of bioluminescence, counter-illumination, alarm displays, decoy flashes, there is a predatory response that exploits or circumvents it. For every predatory use, lures, searchlights, prey illumination, there is a defensive counter-adaptation that detects, avoids, or neutralizes it. The deep ocean is not a static gallery of glowing organisms. It is a battlefield, and the weapons and defenses are made of light. The blue-blind prey of Malacosteus did not choose to be blind to red. They are optimized for the blue wavelengths that dominate deep-sea bioluminescence, which is an excellent adaptation against most predators. But Malacosteus has found the gap in their defenses, a frequency they cannot detect, and exploited it with lethal precision. This is exactly the kind of escalating adaptation that characterizes arms races in biology, and it suggests that the bioluminescent arms race in the deep ocean has been running for a very long time, producing increasingly sophisticated offensive and defensive capabilities with each passing generation. The environmental explanation also does not account for why certain species of deep-sea squid can produce light displays that change color, pattern, and intensity within fractions of a second, creating what researchers have described as whole-body animations, rapid sequences of light that ripple across the animal's skin like text scrolling across a screen. These displays are too complex, too fast, and too variable to be simple predator avoidance responses. They look, to the trained eye of a biologist accustomed to communication behaviors, like language, like information exchange, but communication with whom? And transmitting what content? The speed of these displays is particularly striking. Some deep-sea squid can produce and extinguish bioluminescent pulses in intervals measured in milliseconds, faster than the human eye can track without high-speed cameras. The patterns that emerge are not random, they repeat, they vary systematically, they differ between individuals, between situations, and between species. A squid that is foraging produces different patterns from a squid that is being approached by a potential mate, and both differ from a squid that has detected a predator. If these patterns are communicative, if they carry information that other organisms can decode, then we are looking at a visual communication system of remarkable bandwidth, a system that can transmit complex, rapidly changing information through an environment where no other form of long-distance communication is available. In the deep ocean, there is no sound channel efficient enough for the kind of rapid, directional information transfer that light permits. There is no chemical signaling system fast enough to match the millisecond scale changes in a squid's photophore display. Light is the only medium that combines the speed, the directionality, and the information density needed for real-time, complex communication in the dark. It does not explain why the deep-sea siphonophore Arena, a colonial organism related to the Portuguese man-of-war, uses bioluminescent lures that glow red and twitch, in a way that convincingly mimics the movement of a small copepod, a common deep-sea crustacean that serves as food for many fish. This is a predator using bioluminescence to create a sophisticated imitation of prey, a fishing lure designed by natural processes to glow in a color that should, according to our understanding, be invisible to most deep-sea creatures. Yet fish are attracted to it, they strike at it, they are consumed. This implies that the target fish can see red light, which contradicts the long-held assumption that most deep-sea organisms are effectively red-blind. Something is wrong with our assumptions, and the siphonophore Arena is one of the most compelling pieces of evidence that our model of deep-sea vision is incomplete. The chemistry of bioluminescence adds yet another layer of complexity. The core reaction, luciferin plus luciferase plus oxygen, is deceptively simple in principle. But the luciferins used by different organisms are not the same molecule. They are chemically distinct compounds that happen to perform the same function. There are at least 4 major types of luciferin found in the ocean. Coelenterrazine, used by an enormous range of organisms from jellyfish to fish to shrimp. Vargolin, used by certain ostracods and the midshipman fish. Dinoflagellate luciferin, structurally related to chlorophyll, used by the glowing plankton that illuminate tropical bays, and bacterial luciferin, a flavin compound used by luminous bacteria and the organisms that host them in their bodies. These are different molecules, with different structures, different synthesis pathways, and different enzymatic partners. They are not variants of a single ancestor molecule. They appear to have arisen independently, through separate biochemical pathways, in organisms that share no recent common ancestry. Coelentrazine alone is found in at least 9 different phyla. Major branches of the animal kingdom that diverged from each other hundreds of millions of years ago, according to current scientific models. This raises a difficult question. Did each of these widely separated lineages independently stumble upon the ability to synthesize coelentrazine through sheer biochemical coincidence, or did they acquire it from an external source? Evidence accumulated over the past 2 decades suggests that many organisms cannot synthesize coelentrazine at all. They obtain it from their diet, eating other organisms that contain it and repurposing the molecule for their own bioluminescent reactions. This means that coelentrazine may function as a kind of deep-sea vitamin, an essential substrate that circulates through the food web, passed from organism to organism, each species using it as the fuel for its own unique light display. If this dietary hypothesis is correct, then the entire deep-sea bioluminescent ecosystem is chemically linked in a way that is only now becoming clear. The light produced by a jellyfish at 3,000 feet may depend on coelentrazine that was originally synthesized by a bacterium at 6,000 feet, eaten by a copepod, consumed by a shrimp, and passed up through the food chain. Cut off the supply of coelentrazine at the base, and the lights would go out, not in one species, but across thousands of species, simultaneously across every ocean basin. The bioluminescent world is not a collection of independent light sources. It may be a single, interconnected, chemically dependent network. This chemical interdependence has consequences that extend far beyond the organisms themselves. If bioluminescence depends on a shared substrate that circulates through the food web, then anything that disrupts the food web can disrupt bioluminescence. Overfishing of mesopelagic species, deep-sea mining operations that destroy benthic habitats, climate-driven changes in ocean temperature and circulation that alter the distribution and productivity of phytoplankton at the surface. All of these factors could, in theory, reduce the supply of coelentrazine in the deep ocean, dimming the bioluminescent displays of organisms that have relied on it for countless generations. We do not know how sensitive the system is. We do not know how much disruption it can absorb before the lights begin to fail. And we do not know what the ecological consequences would be if a significant fraction of deep-sea organisms lost their ability to glow. If bioluminescence is as fundamental to deep-sea survival as the evidence suggests, then disrupting it could cascade through the ecosystem in ways we cannot predict. It would be like removing language from human society. The structures that depend on it, trade, cooperation, defense, courtship, would collapse, and the web of chemical dependence may extend even further than coelentrazine. Recent research has identified other molecules that appear to circulate through deep-sea food webs and serve as essential components of bioluminescent reactions. Fluorescent proteins, which absorb the blue light produced by the luciferin-luciferase reaction and re-emit it at longer wavelengths, shifting it from blue to green, yellow, or even red, are found in a wide range of deep-sea organisms. Some of these proteins may also be obtained from the diet rather than synthesized internally, adding another layer of chemical interdependence to the bioluminescent network. The deep-sea light show is not powered by individual batteries. It is powered by a shared electrical grid, and we do not yet know where all the cables run. But here is what makes this truly terrifying. We are not even close to a complete inventory. The deep ocean remains the least explored major habitat on Earth. Scientific estimates suggest that fewer than 5% of the deep-sea floor has been directly observed by human eyes or cameras. The water column above it, the vast three-dimensional space where most bioluminescent organisms live, hunt, migrate, and communicate, is even less well surveyed. Every major deep-sea expedition discovers new species. Every deployment of a new observation technology reveals organisms and behaviors that were previously unknown. In 2020, a team from the Schmidt Ocean Institute, using a remotely operated vehicle in the Indian Ocean, filmed a siphonophore that measured an estimated 150 feet in length, potentially the longest animal ever recorded. It was bioluminescent. It had never been seen before. The bioluminescent world beneath 3,000 feet is not a solved puzzle with a few missing pieces. It is a puzzle where we have found only the corner pieces and the edge fragments, and we are staring at a vast, dark interior that may contain patterns, connections, and structures we cannot yet imagine. We know enough to know that something immense is happening in the darkness. We do not know enough to say what it is. And every time we develop a new tool, a new technique, or a new approach to observing the deep ocean, the puzzle does not shrink. It grows. The more we see, the more we realize how much we have been missing. The mystery of deep-sea bioluminescence is not becoming simpler with more data. It is becoming more complex. And the gap between what we know and what there is to know is not closing. It is widening, with each new discovery revealing not just a new answer, but an entirely new category of question that we did not know to ask. Humans have tried, in several distinct ways, to penetrate the bioluminescent world of the deep ocean. Each attempt has revealed something extraordinary. Each has also exposed the severe limitations of our technology, our understanding, and our assumptions about what lies below. What follows is the account of 5 different approaches to this problem. 5 windows into the glowing abyss, and what each one has shown us about a world we still cannot fully see. The first window was opened by the hydrothermal vents. In 1977, a team of geologists aboard the research submersible Alvin descended to a depth of approximately 8,000 feet near the Galapagos Rift in the eastern Pacific Ocean. They were not looking for life. They were looking for geological features, cracks in the ocean floor where superheated water, driven by volcanic activity deep within the Earth's crust, was believed to escape into the surrounding cold seawater. What they found when they reached the bottom changed the course of biology. Clustered around the vents were dense communities of organisms that, according to everything scientists believed at the time, had no business being there. Giant tube worms, some reaching over 6 feet in length, swayed gently in the current. Their bodies were encased in white, chitinous tubes, and their feathery, blood-red plumes extended into the warm water pouring from the vents. Clams the size of dinner plates lined the rocky crevices. Dense mats of white bacterial filaments draped over the vent structures like snow on a rooftop. Pale crabs and swarms of shrimp scuttled across the rocks. The scene was not a barren wasteland. It was an oasis, a thriving ecosystem at a depth where, by all prior understanding, the food supply should have been far too scarce to support such abundance. The explanation came quickly and rewrote the textbooks. These organisms did not depend on sunlight. They depended on chemistry. The bacteria at the base of the vent food web were performing chemosynthesis, oxidizing hydrogen sulfide and other reduced chemicals pouring from the vents, using the energy released to fix carbon dioxide into organic molecules. This was photosynthesis without the photo. It was life powered by the heat of the Earth itself, independent of the sun, independent of the surface, independent of everything that biologists had assumed was necessary for complex ecosystems to exist. And many of these vent organisms, researchers would later discover, were bioluminescent. The shrimp that swarmed around the vents, particularly species of the genus Rimicaris, possessed peculiar structures on their backs, not true eyes, but light-sensitive patches that appeared to detect the extremely faint glow emitted by the superheated vent water itself. This glow, known as thermal radiation or blackbody radiation, is so incredibly dim that it is invisible to the human eye even in total darkness. The human retina simply lacks the sensitivity to detect it. But the vent shrimp had developed organs specifically tuned to these wavelengths, presumably to navigate safely near the scalding vent fluid. Swimming too close meant death by boiling. The shrimp needed to know where the hottest water was, and they used their photoreceptors to find it. They were seeing heat, and some researchers speculated that the faint thermal glow of the vents might have been one of the earliest light sources that deep-sea organisms learned to detect, a primordial stimulus that eventually drove the development of more sophisticated eyes and, ultimately, the ability to produce light artificially through bioluminescence. Other vent organisms produce their own light as well. Bioluminescent fish lurked at the periphery of vent fields, presumably attracted by concentrations of prey. Certain species of octopus observed near vents displayed photophore activity, And the dense bacterial mats that covered the vent surfaces included bioluminescent species whose faint, continuous glow may have served ecological functions that are not yet understood, possibly attracting specific organisms that graze on bacterial mats, or signaling the chemical composition of the surrounding water to organisms capable of interpreting such information. The vent discovery carried a secondary implication that would take years to fully appreciate. Before 1977, it was taken as a given that the sun was the ultimate engine of all biology on Earth. Every food chain, every ecosystem, every known living organisms depended, directly or through intermediaries, on the energy of solar photons captured by photosynthetic organisms and passed upward through food webs. Chemosynthesis shattered that assumption. Here was a complete ecosystem, primary producers, grazers, predators, scavengers that owed nothing to the sun. Not a single calorie in the vent community had passed through a chloroplast. Not a single photon of sunlight had contributed to the construction of any organism in the system. The energy came from below, from the planet itself, from chemical reactions between superheated rock and ocean water that would continue for as long as the Earth retained its internal heat. The implications for bioluminescence were immediate and troubling. If life could flourish without sunlight, using chemical energy from the Earth's interior, then what role did light, the production of visible photons by living cells, play in an ecosystem that had no relationship with photons from any external source? The vent organisms that bioluminesced were not mimicking sunlight. They were not compensating for the absence of the sun. They were producing light in a place where light had never existed, for organisms that had never experienced illumination from any source other than their own bodies and those of their neighbors. The light was entirely self-referential, generated and consumed within a community that had no concept of any other kind of light. If bioluminescence could evolve and persist in an ecosystem that had never known the sun, then perhaps the common assumption that deep-sea bioluminescence is a response to the loss of sunlight was incomplete. Perhaps bioluminescence is not a substitute for the sun. Perhaps it is something else entirely, something more fundamental, something that life in the dark develops not because it misses sunlight, but because light itself, regardless of its source, is intrinsically useful in ways we have not fully catalogued. But here is the problem with the hydrothermal vent window. Vents are isolated. They occur along mid-ocean ridges and in back-arc basins, narrow, geologically active strips of the ocean floor where tectonic plates are spreading apart or colliding. The total area of the ocean floor covered by active hydrothermal vent fields is vanishingly small compared to the total seabed. The bioluminescence found at vents, while spectacular and biologically significant, represents a small and specialized subset of the total phenomenon. It does not explain why bioluminescence dominates the open water column, thousands of miles from any vent, in the vast midwater zones that make up the bulk of the deep ocean's living space. The vents are important, they are illuminating, but they are a footnote, not the main text. The second window was opened by brine pools. Deep-sea brine pools are among the most surreal features anywhere on the ocean floor. They are, in the most literal sense, lakes at the bottom of the sea. Dense, hypersaline bodies of water that sit in depressions on the ocean floor, like ponds in a landscape, complete with distinct shorelines, visible surfaces, and in some cases even waves that ripple across the brine-seawater interface when disturbed by currents. The brine is so dense, sometimes 5 to 8 times saltier than normal seawater, that it sinks to the lowest available point and stays there, refusing to mix with the overlying water column. The interface between the brine and the seawater above is sharp, clearly visible, and strangely beautiful. Submersible cameras have filmed this boundary as a shimmering, mirror-like surface, an underwater beach where the water meets an even denser water. It creates the visual illusion of a shoreline 2 miles below the surface of the ocean. Brine pools are toxic to most marine life. The combination of extreme salinity, very low dissolved oxygen, and high concentrations of methane and hydrogen sulfide creates a chemical environment that is lethal to the vast majority of fish, crustaceans, and invertebrates that venture too close. The shores of brine pools are often littered with the preserved remains of organisms that strayed into the brine and could not escape. Dead crabs, dead fish, dead eels, their bodies pickled by the salt, lying in rows along the edge of the pool like the casualties of an invisible battle. It is a deep-sea graveyard, and it is one of the most haunting sights in the ocean. And yet, at the margins of these pools, where the brine meets the normal seawater in a thin, transitional layer, specialized communities thrive. Dense mats of chemosynthetic bacteria line the edges, feeding on the chemical energy dissolved in the brine. Mussels cluster along the boundary in tight aggregations, their tissues hosting symbiotic bacteria that process the toxic sulfide and methane into usable energy. Tube worms anchor themselves at the very edge of the pool, their plumes reaching into the chemical gradient where the concentrations of their required substrates are highest. And polychaete worms, small crustaceans, and other invertebrates move along the shore, grazing on the bacterial mats or filtering particles from the transitional water. Here, too, bioluminescence appears. The organisms living near brine pools produce light, though the specific functions of that light are not fully understood. Some researchers believe the light may serve as a warning signal, a beacon to other organisms that the area ahead is dangerous. Others have suggested that bioluminescent bacteria within the brine itself may be responsible for some of the observed glow, producing light as a byproduct of their metabolic processes or as a quorum sensing response when their population density reaches a critical threshold. Still others have proposed that bioluminescence near brine pools may be involved in attracting specific prey organisms to the chemically rich margins, where predators wait. The brine pool environment is so extreme, so geographically isolated, and so poorly studied that almost any hypothesis about its bioluminescence remains plausible. What is certain is that even in one of the most hostile chemical environments on the planet, a place where the water itself is lethal, life has found a way to persist, and that life glows. What makes the brine pool window particularly valuable is not the bioluminescence itself, but what it reveals about the relationship between toxicity and light. The margins of brine pools are chemical boundaries, sharp gradients where oxygen, salinity, temperature, and dissolved gas concentrations change dramatically over distances measured in inches. Organisms that survive at these margins must maintain extraordinarily precise control over their own internal chemistry, regulating the flow of ions, managing dissolved gases, and preventing the toxic brine from overwhelming their cells. The fact that these same organisms also devote metabolic resources to bioluminescence is striking. In an environment where every biochemical process is strained to its limit by the surrounding conditions, the production of light is apparently not a dispensable luxury. It is maintained alongside the most fundamental survival processes: osmoregulation, respiration, waste management, suggesting that its function is equally fundamental. Some researchers have speculated that bioluminescence at brine pool margins may be linked to chemical sensing rather than visual communication in the conventional sense. The organisms that live at these boundaries need to monitor the chemical gradient constantly. Too close to the brine, and they die. Too far, and they lose access to the chemical energy that sustains them. Light production in certain bacteria is known to respond to specific chemical concentrations, brightening or dimming as the surrounding chemistry shifts. If the organisms at brine pool margins use bioluminescence as a chemical indicator, a real-time visual readout of the conditions in their immediate environment, then they are using light not to communicate with other organisms, but to monitor their own position relative to a lethal boundary. They are using light to stay alive in a more direct and immediate sense than any predator-prey or mating signal. But again, brine pools are localized features. They are found in specific geological settings: the Gulf of Mexico, the Red Sea, the Eastern Mediterranean, certain areas of the Arctic, and they represent a tiny fraction of the ocean floor. The bioluminescence observed at brine pools, like that at hydrothermal vents, is spectacular in isolation but does not address the broader mystery. The midwater column, where most bioluminescent organisms live, is thousands of miles from any brine pool. The forces shaping bioluminescence in the open ocean must be different from those at work in these extreme, localized environments. The third window came through the study of milky seas and the strange optical phenomena of the open ocean. For centuries, sailors have reported eerie lights in the water, glowing seas that stretch to the horizon, milky luminescence that seem to emanate from the depths, strange underwater flashes beneath the hull during calm, dark nights. These reports were long dismissed by scientists as superstition, optical illusions, phosphorescence, a catch-all term that was used loosely and often inaccurately to describe any unexplained light on the water. But in recent decades, satellite imagery and modern oceanographic instruments have confirmed that some of these reports were describing real phenomena of enormous scale. In 2005, scientists at the Naval Research Laboratory and collaborators at the Monterey Bay Aquarium Research Institute published a landmark analysis of satellite data from a region of the Indian Ocean where a British merchant vessel, the SS Lima, had reported sailing through a vast, uniformly glowing sea in 1995. The crew of the Lima described the ocean as glowing a pale, milky white from horizon to horizon, lasting for several hours as the ship moved through the area. The glow was uniform, steady, and extended in every direction. It was not patches of phosphorescence. It was not reflected moonlight. It was the ocean itself glowing. The satellite data confirmed the account. A massive bioluminescent event had indeed occurred in the region at the time and date of the Lima's report. The glowing area, as measured from space, was estimated to cover approximately 6,000 square miles, an expanse larger than the state of Connecticut, visible from orbit and maintained for multiple days. The numbers are striking. 6,000 square miles of ocean, producing a uniform, continuous pale glow in the middle of the night, far from any coastline, any human settlement, any artificial light source. It was one of the largest coordinated biological displays ever documented. The confirmation of the Milky Sea from space was a turning point in how scientists thought about bioluminescence at scale. Individual organisms producing light, a flashing jellyfish, a glowing squid, are fascinating but manageable. They can be studied in laboratories, described in papers, and incorporated into existing models of deep-sea ecology without requiring any fundamental revision of how we think about ocean systems. But a bioluminescent event visible from orbit is something else entirely. It is not a specimen, it is a phenomenon. It is a biological process operating at a scale that more closely resembles weather than behavior, a living light event covering an area that would take a ship days to cross, involving a number of individual organisms that exceeds any meaningful human intuition about quantity. The Milky Sea is not a large bioluminescent display, it is a bioluminescent climate event, a manifestation of biological light production at a scale where the appropriate comparison is not a firefly, but a thunderstorm. Subsequent analysis of historical satellite data revealed that Milky Sea events, while rare, are not unique. Multiple events have been identified in the Indian Ocean, the Arabian Sea, and other regions, typically in warm tropical waters where nutrient conditions favor massive bacterial blooms. Some of these events lasted for days, others persisted for weeks. The total number of Milky Sea events that have occurred in the world's oceans since satellite coverage began is not known with precision, because many occurred before researchers knew what to look for in the data. It is entirely possible, even likely, that Milky Seas occur more frequently than the current catalogue suggests, in regions where satellite coverage is sparse, or where the events coincided with cloud cover that blocked the sensors. The leading explanation for these Milky Seas involves enormous blooms of bioluminescent bacteria, trillions upon trillions of individual cells, each producing a faint but continuous glow, collectively creating a light source so vast that it can be detected by satellites hundreds of miles above the Earth. The species most commonly implicated is Vibrio harveyi, a bioluminescent marine bacterium that is known to engage in quorum sensing, a chemical communication system in which individual bacteria release signaling molecules, called autoinducers, into the surrounding water. When the concentration of these molecules reaches a certain threshold, indicating that the bacterial population has achieved a critical density, every bacterium in the bloom simultaneously activates its bioluminescence genes. They do not glow individually. They glow collectively in a coordinated population-wide response that creates the uniform milky radiance observed from both sea level and space. The implications of quorum sensing in the deep ocean are staggering and largely unexplored. If surface water bacteria can coordinate bioluminescence across thousands of square miles using chemical signals, what is happening at depth? Bioluminescent bacteria are far more prevalent in the deep ocean than at the surface. They colonize the guts, the skin, and the specialized light organs of countless deep-sea organisms. They form biofilms on marine snow particles as they drift downward through the water column. They coat the surfaces of dead organisms that settle on the abyssal floor. Are these deep water bacteria also engaging in quorum sensing? Are there coordinated bioluminescent events happening at depth? Massive, synchronized light displays in the midnight zone that no camera has ever recorded. The quorum sensing question leads to an even deeper uncertainty about the nature of coordination in the deep ocean. On the surface, bioluminescent bacteria achieve coordination through chemistry. The autoinducer molecules they release into the water serve as a voting system, a way for individual cells to assess the density of their neighbors and respond when a threshold is reached. But in the deep ocean, where water currents are slow, temperatures are stable, and bacterial populations can remain in close proximity for extended periods, the conditions for quorum sensing are in many ways more favorable than at the surface. The molecules have more time to accumulate. The populations are more stable. The chemical signals are less likely to be dispersed by turbulence. If quorum sensing drives bioluminescence in the relatively chaotic surface waters, it may drive even larger, even more sustained, even more precisely coordinated events in the stillness of the deep. There is another dimension to this possibility that is rarely discussed but worth considering. Quorum sensing is not merely a switch that turns bioluminescence on or off. Research in microbiology has shown that quorum sensing regulates dozens of genes simultaneously. Genes involved in virulence, biofilm formation, antibiotic production, and nutrient acquisition. In bioluminescent bacteria, the quorum sensing system that activates light production may also activate other systems that alter the behavior and metabolism of the bacterial community in ways we have not measured in deep-sea populations. The light may be only the visible symptom of a much broader coordinated response, a bacterial consensus that changes the chemistry, the toxicity, and the ecological impact of an entire microbial community simultaneously. When trillions of bacteria in the deep ocean reach quorum and begin to glow, they may also be changing the chemical environment around them in ways that affect every organism in the vicinity. The light is what we see. What we do not see may be equally important. We do not know. The Milky Sea phenomenon has been confirmed for surface waters, but its deep water equivalent, if it exists, has never been observed. The possibility alone is enough to reshape our understanding of what the deep ocean might look like if we could see it in its entirety. But the Milky Sea window, like the others, has its limitations. These events are episodic and unpredictable. They appear to be triggered by specific combinations of nutrient availability, water temperature, bacterial population density, and other factors that are not fully characterized. They tell us that coordinated bioluminescent behavior is possible on a massive scale, but they do not explain the daily, routine, individual bioluminescence of the fish, squid, jellyfish, and worms that populate the deep water column. The fourth window was opened by the discovery of creatures that appear to defy the physical constraints of their environment. The deep ocean is a place of extremes: extreme pressure, extreme cold, extreme darkness, and extreme scarcity of food. Organisms living there are expected to be small, slow, energy efficient, and conservative in their use of resources. Every metabolic expenditure must be justified by a clear survival benefit, because there are no calories to spare. And yet, bioluminescence, which requires metabolic energy to produce, specialized organs to house, and complex chemistry to maintain, appears to be anything but conservative. Some of the most extravagant, energy-intensive displays of bioluminescence occur in the deepest, most resource-starved environments on the planet. The deep-sea jellyfish Atolla wivillae, sometimes called the alarm jellyfish, produces one of the most dramatic bioluminescent displays in the ocean. When attacked by a predator, Atolla does not simply flash once and go dark. It generates a rotating spiral of brilliant blue light that spins around its bell like a pinwheel or a police siren, creating a visual alarm signal that can be seen from hundreds of feet away through clear, deep ocean water. This display lasts for several seconds, an eternity in a predator-prey encounter, and its purpose, as demonstrated by Edith Widder's experiments with the electronic jellyfish lure, is to attract larger predators to the scene. Atolla is screaming for help, writing its distress call in spinning blue fire. The Atolla alarm is remarkable not only for its effectiveness, but for its specificity. Researchers have analyzed the rotation speed, intensity, duration, and spectral characteristics of the display and found that it is not random. The spiral rotates at a consistent speed. The intensity follows a predictable curve, starting bright and gradually diminishing as the jellyfish depletes its supply of luminescent substrate. The wavelength is tightly clustered around 470 nanometers, the blue wavelength that travels farthest through seawater and matches the peak sensitivity of most deep-sea visual systems. Every parameter of the display appears to be optimized for maximum visibility over maximum distance in the specific medium of deep ocean water. The alarm is not a crude flash. It is a precisely engineered signal, tuned to its environment with a degree of precision that would be impressive in a telecommunications system designed by engineers. And it is produced by a jellyfish, an organism without a brain, without a central nervous system, without any structure that we would recognize as a command center capable of designing and calibrating such a display. The sophistication of the output vastly exceeds what the simplicity of the organism would seem to allow. But the energy cost of this display is not trivial. Atolla lives at depths where food particles drift down from above in quantities measured in milligrams per square meter per day. Every calorie the jellyfish consumes is precious. Every molecule of luciferin it expends on light production is a molecule it cannot use for growth or reproduction. And yet, Atolla has devoted a significant portion of its metabolic budget to producing a light show that it may deploy only a handful of times in its entire lifespan. The fact that this display persists, that natural processes have not eliminated it, is powerful evidence that the survival benefit of the alarm display outweighs its considerable metabolic cost. The bioluminescent alarm works, it saves lives, and the deep ocean is a place where being able to call for help in the dark can mean the difference between being eaten and living to reproduce. The deep-sea octopus, Stauroteuthis satensis, provides another example of bioluminescence in an organism that, by all expectations, should be conserving every possible resource. This is one of only a few octopus species known to be bioluminescent, and its adaptation is particularly striking because it involves a fundamental trade-off. Most octopuses use their suckers, the muscular, adhesive discs that line their arms, for gripping surfaces, manipulating objects, and capturing prey. Suckers are one of the most versatile tools in the octopod body plan. But in Stauroteuthis, the suckers have been modified into photophores. Instead of the typical muscular suction cups, this octopus has rows of light-emitting organs that produce a steady greenish-blue glow. The suckers no longer grip, they shine. This is a significant sacrifice. The octopus has traded one of its most useful physical tools for the ability to produce light. The implications of this trade extend beyond the individual organism and speak to the evolutionary pressures operating in the deep ocean. Octopuses are among the most intelligent and adaptable invertebrates in the ocean. Their suckers are not simply passive adhesive discs. They are sensory organs, rich in chemoreceptors and tactile nerve endings, capable of tasting and feeling the objects they touch. In shallow water octopuses, the suckers are integral to nearly every aspect of the animal's life: hunting, navigation, exploration, self-defense, manipulation of tools, and even social interaction. For an octopus to abandon this sensory and mechanical capability in favor of photophores represents a trade so extreme that it could only have occurred under intense selective pressure so strong that in the deep-sea environment, the ability to produce light outweighed the combined advantages of grip, taste, and touch. The function of this light is debated among researchers. It may attract small copepods and other planktonic prey toward the octopus's web and arms, where they can be trapped and consumed. It may serve as a display during mating encounters, allowing individuals to identify each other and assess fitness in the dark. It may have a defensive function that has not yet been observed. What is clear is that the trade was considered worthwhile, that in the deep ocean, the ability to produce light is more valuable than the ability to grip. Light is worth more than touch. Perhaps the most unsettling example of bioluminescent extravagance is the black dragonfish, Idiocanthus atlanticus. The females of this species are formidable predators, reaching lengths of up to 16 inches, with elongated, serpentine bodies, massive jaws lined with fang-like teeth, and rows of photophores along their bellies and behind their eyes. They are among the most efficient hunters in the deep sea, using their bioluminescence to illuminate prey, to camouflage their silhouettes through counter-illumination, and possibly to communicate with other dragonfish at a distance. But the males of the species tell a different story. Adult male black dragonfish are tiny, often less than 2 inches long, with no functional teeth, no functional digestive system, and no ability to eat. Their jaw structures degenerate. Their stomachs shrink and cease to function. Once a male reaches maturity, he does not feed. He cannot feed. He lives off stored energy reserves accumulated during his larval stage, and those reserves are finite. His adult life is a countdown. He has only enough energy to search for a female mate and die. His entire purpose is reproduction, and his entire adult existence is measured in weeks, not years. And yet, throughout his brief adult life, the male black dragonfish continues to bioluminesce. He still produces light. Even as his body is consuming its own tissues for energy, even as his organs are shutting down one by one, even as death approaches with absolute certainty, the photophores continue to glow. This raises a haunting question that researchers have not fully resolved. What is the light for? Is it a beacon to females, a mating signal that says, "Here I am, come find me"? Is it a vestige of a developmental program that activates the photophores during the larval stage and simply never turns them off? Or is bioluminescence so deeply embedded in the biology of these organisms, so woven into the fabric of their cells and tissues, that it continues even when there is no longer any survival benefit, even when the organism producing it is already in the process of dying? The male dragonfish raises a question that extends far beyond his own species, and into the broader nature of bioluminescence in the deep sea. If light production persists even in an organism that is dying, even when the metabolic cost is being extracted from irreplaceable tissue reserves, then bioluminescence may not be simply an optional behavior that organisms activate and deactivate in response to circumstances. It may be something closer to a fundamental property of the organism's biochemistry, a process so deeply integrated into cellular function that it cannot be switched off without dismantling the cell itself. There are precedents for this kind of deep integration in biology. The human heart does not stop beating because the body decides it would be more efficient to rest. The cells of the heart are organized around the act of contraction, and they continue contracting until they physically cannot. If bioluminescence in some deep-sea organisms is similarly embedded, not an optional feature but a defining characteristic of cellular function, then the question of why the deep ocean glows is not a question about behavior or strategy. It is a question about what life in the deep ocean fundamentally is. And the answer may be that, at the deepest level, life in the deep ocean is light. Not life that produces light, but life that cannot exist without producing it. The fourth window also reveals a pattern that is easy to overlook in the details of individual species, but becomes unmistakable when the examples are considered together. Each of these organisms, the alarm jellyfish, the sucker-to-photophore octopus, the self-consuming dragonfish, has sacrificed something significant for bioluminescence. Atolla devotes precious metabolic reserves to a display it may use only a few times. Storoteuthis has given up functional suckers. The male dragonfish burns through his final energy reserves to keep his photophores alight. In each case, the organism has made a trade, and in each case, the trade favors light over some other capability. The deep ocean is a place where light is so valuable that organisms will give up grip, give up food, give up life itself to produce it. There is no comparable sacrifice on the surface. No land animal gives up a limb for the ability to glow. No bird trades its feathers for photophores. The asymmetry speaks to something fundamental about the deep sea environment. In absolute darkness, light is not merely useful, it is worth almost any price. The fifth window, and in many ways the most revealing, was opened by the development of new observational technologies that allow researchers to observe the deep ocean without disrupting it. The eye in the sea was the pioneer, but it was not the last. The Medusa camera system, also developed by Edith Widder's team at the Ocean Research and Conservation Association, uses far-red illumination that is invisible to most deep-sea eyes, combined with bioluminescent attractors that mimic natural deep-sea signals. The system can be deployed on the seafloor for days at a time, recording continuously, waiting for the inhabitants of the deep to reveal themselves on their own terms. These cameras have captured phenomena that no one predicted, and that would have been invisible to any traditional observation method. The significance of these unlit, passive observation systems cannot be overstated. For decades, every deep-sea instrument had been designed around the assumption that the observer must bring light into the darkness. Powerful white floodlights, strobes, laser illumination, in order to see. The Medusa and Eye in the Sea systems inverted that logic entirely. Instead of overwhelming the darkness with artificial light, they embraced it. They sat in the dark and waited for the organisms to produce their own light. And the organisms obliged, revealing behaviors, species, and ecological interactions that had been invisible to every brightly lit camera and submersible that had come before. The philosophical implication of this approach deserves emphasis. The deep ocean had been telling us, through decades of underwhelming survey results and suspiciously sparse trawl catches, that our methods were the problem. But it took a leap of imagination. Widder's decision to stop looking with human eyes adapted to sunlight and start looking with instruments adapted to bioluminescence. To prove it, the deep ocean was not empty, it was hiding, and it was hiding specifically from the tools we had designed to find it. Our instruments, built to illuminate a dark world, were functioning as the world's most effective wildlife repellents. In 2012, a Medusa camera deployed in the Gulf of Mexico at a depth of approximately 3,000 feet captured footage of a giant squid, Architeuthis dux, attacking the bioluminescent lure. This was among the first recordings of a living giant squid in its natural deep-sea habitat. The animal was not attracted by food. It was not attracted by sound, by chemical cues, or by vibration. It was attracted by light. A pattern of blue flashes, mimicking the alarm display of a jellyfish, drew one of the ocean's most elusive predators out of the darkness and into view of the camera. The giant squid came to investigate a bioluminescent signal, suggesting that it routinely monitors the darkness for such signals, that it hunts by watching for the light produced by other organisms, and that its relationship with bioluminescence is not incidental but central to its predatory strategy. The giant squid footage was a milestone not only because it showed the animal alive in its habitat, but because it demonstrated definitively that the largest and most powerful invertebrate predators in the deep ocean use bioluminescence as a primary hunting cue. If Architeuthis, an animal that can weigh over 600 pounds and reach lengths exceeding 40 feet including its tentacles, responds to bioluminescent signals, then every bioluminescent organism in the deep ocean is participating in a visual ecosystem that includes apex predators of enormous size and power. The light in the deep is not a quiet, inconsequential affair. It is being watched by some of the largest eyes in the animal kingdom, on organisms that can strike with devastating speed across distances of several body lengths. The stakes of bioluminescent communication in the deep ocean are literally life and death, conducted in the presence of predators that most surface-dwelling humans would find difficult to imagine. In other deployments, deep-sea cameras captured images of siphonophores, the immense, colonial organisms related to jellyfish and the Portuguese man-of-war, producing coordinated bioluminescent waves. These were not random, disorganized flickers. They were organized pulses of light, that travelled along the length of the colony, which in some cases measured over 120 feet, propagating in specific directions, at measurable speeds, in repeating patterns. To the researchers who observed them, these waves had the unmistakable appearance of signals being transmitted along a biological cable, information moving from one end of a colonial organism to the other, encoded in light. Whether these waves carry information in any meaningful sense, and if so, what kind of information and to what recipients, remains one of the most compelling open questions in deep-sea biology. But the observation itself is profound. A single colonial organism, longer than a blue whale, pulsing with synchronized light in the absolute darkness of the midnight zone, transmitting patterns that propagate along its body like electrical impulses along a nerve fiber. This is not a simple chemical reaction producing a static glow. This is dynamic, directional, patterned, bioluminescent behavior in an organism that most people have never heard of, in a habitat that most people will never see. The siphonophore light waves raise a question that cuts to the heart of what we mean by communication. Communication, in the biological sense, requires a sender, a signal, a medium, and a receiver. The siphonophore is clearly the sender, the light wave is clearly the signal, the water column is clearly the medium. But who is the receiver? Is the siphonophore communicating with other siphonophores in the vicinity, coordinating behavior across a population of colonial organisms spread through the water column? Is it communicating with itself, sending internal coordination signals along its own body, synchronizing the behavior of thousands of individual zooids that make up the colony? Or is it communicating with organisms of entirely different species, prey that it hopes to attract, predators that it hopes to confuse, or organisms that we have not yet observed responding to the signal? The answer matters because it determines whether we are looking at a reflex or a conversation. If the light waves are internal coordination signals, the siphonophore equivalent of nerve impulses, then they are impressive but not fundamentally different from the electrical signals that coordinate muscle contractions in a fish or the heartbeat of a mammal. But if the light waves are external signals directed at other organisms, then we are looking at something far more significant: a bioluminescent broadcast, transmitted through the deep ocean, carrying information that other organisms are evolved to receive and interpret. That would make the siphonophore not just a glowing animal, but a broadcaster, a biological transmitter operating in an optical frequency band that we are only beginning to tune into. Other deployments have captured equally startling footage. Cameras positioned near the seafloor at abyssal depths have recorded waves of bioluminescent activity that sweep across the sediment surface like ripples in a pond, as though something below or within the sediment triggered a chain reaction of light production in the bacterial communities and buried organisms that inhabit the upper layers of the abyssal mud. The cause of these waves is unknown. They are too large and too uniform to be explained by the movement of a single animal disturbing the sediment. They are too coordinated to be random. They behave, in the words of one researcher who observed them, like a signal propagating through a network, as though the organisms in the sediment are connected by something, responding to something, and the light is the visible trace of that response, moving through a community of organisms that we cannot see, buried in mud at the bottom of the ocean. And still, every observation raises more questions than it answers. The cameras see only what passes in front of their lenses. The lures attract only those organisms sensitive to their specific wavelength, intensity, and flash pattern. The vast majority of the deep ocean, the darkness between the cameras, the water beyond the range of the lures, the depths below and above the deployments, remains entirely unseen. We have opened 5 windows into the bioluminescent abyss, and through each one we have glimpsed enough to know that we are looking at something vast, something complex, something that operates according to principles we do not yet fully understand. The bioluminescent world beneath 3,000 feet is not a simple phenomenon with a simple explanation. It is a global system, a communication network, a chemical economy, a predator-prey arms race, an ecological organizing principle, and possibly something more, something that our current scientific frameworks are not yet equipped to describe. And the deeper we look, the wider the mystery becomes, and the more we realize how much remains hidden in the dark. Here is the unsettling reality that emerges when you pull together everything we have discussed. Bioluminescence is not a footnote in the story of life on Earth. It is not a quirk, not an oddity, not a minor adaptation found in a handful of exotic species at the margins of the biological world. It is one of the most widespread biological capabilities on the planet. It dominates the largest habitat on the planet, and we have barely begun to understand it. The numbers bear repeating, because they are difficult to accept even after you have heard them once. The deep ocean, everything below approximately 660 feet, makes up more than 90% of the livable space on Earth. Not 90% of the ocean. 90% of the livable space on the entire planet, when you account for the three-dimensional volume of the water column. From the bottom of the sunlit zone to the deepest trench floor, the deep ocean dwarfs every other habitat in existence. Every forest on Earth, every grassland, every desert, every mountain range, every river system, every freshwater lake, every cave system, every patch of tundra, every coral reef, every urban environment built by human hands, all of it, taken together, stacked and compressed, and measured against the volume of the deep ocean, occupies less than 10% of the planet's livable space. The deep ocean is not one habitat among many. It is the habitat. Everything else is a margin note, and in that habitat, the majority of organisms produce their own light. Let that sink in. The world that most people picture when they think of Earth, sunlit, green, familiar, populated by organisms that see by the light of a star, is the exception. The norm, by volume, by area, and by sheer number of organisms, is dark, cold, under crushing pressure, and illuminated not by sunlight, but by the bodies of living things. The sunlit world is a thin veneer stretched over a planet whose dominant visual experience is biological light. If you could somehow strip away the continents, drain the surface waters, and look at the Earth as it truly is, a sphere dominated by deep ocean, you would see a world defined not by sunshine but by bioluminescence. The living light of the deep is the planet's primary visual currency. We just happen to live in the small, bright corner where it is not needed. And yet, here is what makes this truth even more unsettling. We have explored more of the surface of the Moon than we have of the deep ocean floor. The Moon, approximately 239,000 miles away in the vacuum of space, has been photographed in its entirety, mapped to sub-meter resolution, and sampled by both robotic and crewed missions. Every crater has a name. Every basin has been measured. Every major geological feature has been characterized and catalogued. The deep ocean floor, which lies a few miles beneath the hulls of ships that cross it daily, remains largely unmapped, unphotographed, and unexplored. The best global maps of the ocean floor have a resolution of roughly 1 to 5 kilometers, meaning that any feature smaller than a kilometer or two across is essentially invisible. At that resolution, the Titanic would be a speck too small to see. An entire hydrothermal vent field would be a pixel. A brine pool would not register at all. We have better topographic maps of the far side of the Moon, a place no human has ever visited or even seen directly with unaided eyes, than we have of the bottom of our own ocean. More humans have walked on the surface of the Moon, 12 individuals across 6 Apollo missions, than have descended to the deepest point of the ocean in all of human history. At the time of the most recent crewed expeditions to the Challenger Deep, the total number of people who had personally witnessed the bottom of the Mariana Trench remained fewer than 2 dozen. Fewer humans have seen the deepest point on their own planet than have left the atmosphere of the Earth entirely and stood on the surface of another world. Let that sink in. The disparity is not merely numerical. It reflects a deep asymmetry in how human civilizations have allocated their curiosity, their resources, and their imagination. We have built radio telescopes that can detect the faint electromagnetic whispers of galaxies billions of light years away. We have launched probes that have exited the solar system, crossing the heliopause into interstellar space. We have constructed particle accelerators that recreate the conditions of the first fractions of a second after the origin of the universe. These are extraordinary achievements. They represent the pinnacle of human ingenuity, directed outward, upward, into the cosmos, but downward, into the ocean that covers 71% of our own planet's surface. The investment has been a fraction of what has been devoted to space. The budget of a single space agency in a single year often exceeds the cumulative global spending on deep-sea research over the past decade. The consequences of this imbalance are written in the blank spaces of our knowledge about the deep ocean. This is not because we lack the desire to explore the deep. It is because the deep ocean is, in many practical respects, harder to reach and harder to survive in than outer space. An astronaut in a spacesuit faces one atmosphere of pressure difference: the pressure inside the suit versus the vacuum outside. The engineering challenge is to keep the air in. A diver or submersible pilot at the bottom of the Mariana Trench faces over 1,000 atmospheres of pressure difference. The engineering challenge is to keep the water out while preventing the vessel from being crushed like a tin can under the heel of a boot. The materials science, the structural engineering, and the life support requirements for deep ocean exploration are, in many ways, more demanding than those for space travel, and the cost reflects this. A single deep-sea submersible capable of reaching Hadal depths costs tens of millions of dollars to build, maintain, and operate. The number of such vessels in the world can be counted on the fingers of one hand. The result is that the bioluminescent world, the world of living light that covers most of the planet's livable volume, remains one of the least studied, least documented, and least understood environments on Earth. We are more familiar with the surface geology of Mars than with the water column at 4,000 feet in the mid-Atlantic. We have higher resolution images of the rings of Saturn than of the abyssal plains beneath the Southern Ocean, and we have devoted more cumulative scientific effort to studying the light of distant stars, hundreds of light years away, than to studying the light produced by organisms living beneath the keels of fishing boats. This is not science fiction. This is the current state of human knowledge about our own world. The philosophical implications of this ignorance deserve careful consideration. Human civilizations have always defined themselves by what they believe they know. Every era has assumed that it understood the essential structure of the natural world, that the major features had been identified, the major processes explained, the major inhabitants catalogued, and repeatedly, that assumption has been proven wrong. The discovery of microscopic life by Antony van Leeuwenhoek in the 17th century revealed an entire world of organisms that no one had suspected existed. The discovery of bacteria, protozoa, and the teeming microbial ecosystems of soil, water, and the human body shattered the assumption that the visible world was the complete world. The discovery of deep-sea hydrothermal vents in the 1970s revealed an entire category of ecosystem that operated without sunlight, challenging the foundational assumption of biology that all life ultimately depends on solar energy. Each of these discoveries exposed not just new facts, but new categories of ignorance, entire dimensions of the natural world that had never been imagined, let alone investigated. And each one forced a painful recalibration of humanity's sense of its own understanding. We thought we knew what was alive. We were wrong. We thought we knew what life needed. We were wrong. We thought we knew the limits of where life could exist. We were wrong. Bioluminescence in the deep ocean is another such discovery, and its implications are still rippling outward through the scientific community. It does not simply add a few new species to the catalogue of life on Earth. It reveals that the dominant form of visual experience on this planet is not sunlight, it is biological light. The majority of eyes on this planet, the majority of photoreceptors, the majority of visual systems, the majority of optical processing organs are not calibrated to detect photons from the sun. They are calibrated to detect photons produced by other living things, in the darkness, under pressure, in the cold. The visual world that we consider normal, the world of daylight and color, and landscapes illuminated by a star 93 million miles away, is a minority experience. Most of the visual activity on this planet takes place in a world we cannot see, in a language of light we cannot interpret. Consider what this means for our understanding of ecology. The textbook model of an ecosystem begins with the sun. Solar energy is captured by photosynthetic organisms, plants on land, phytoplankton in the ocean, and converted into chemical energy that flows through the food web via herbivores, predators, and decomposers. This sun-driven model has been the central framework of ecology for more than a century. It is the first thing taught in every introductory biology course. It is the foundation of our understanding of how energy moves through living systems. But in the deep ocean, there is no sunlight. The primary energy source for most deep-sea organisms is the rain of dead organic material from above. Marine snow, the constant, slow drift of corpses, fecal pellets, shed scales, and other organic debris from the productive surface waters. This energy input is meager by surface standards. The deep ocean receives perhaps 1 to 5% of the organic matter produced at the surface. It is, by any conventional measure, an energy-poor environment, and yet it is filled with organisms that spend precious metabolic energy on producing light. This is not a trivial expenditure. Bioluminescence requires luciferin, which must either be synthesized internally or obtained from the diet. It requires luciferase or photoprotein enzymes, which must be manufactured by the organism's own cells. It requires specialized organs, photophores, that must be grown, vascularized, innervated, and regulated in an environment where every calorie is precious and every metabolic shortcut confers a survival advantage. The fact that so many organisms devote substantial resources to light production is a powerful argument that the benefits of bioluminescence are immense. It is not a decoration. It is not an accident. It is a survival necessity so critical that organisms will sacrifice other capabilities, grip, tissue, digestive function, to maintain it. The metabolic investment in bioluminescence also has implications for how we think about energy budgets in the deep ocean. The total amount of chemical energy devoted to light production across all bioluminescent organisms in the deep ocean has never been measured. It may be unmeasurable with current technology, given the number of organisms and the inaccessibility of the environment. But the calculation, even in rough form, is illuminating. If the deep ocean contains, conservatively, billions of tons of bioluminescent organisms, and if each organism devotes even a small fraction of its metabolic output to light production, the total energy flux represented by bioluminescence is enormous, a planetary-scale biochemical phenomenon that converts chemical energy into photons continuously in every ocean at every depth below the sunlit zone. This energy does not come from the sun. It comes from the food web, from the organic matter that rains down from the surface, from chemosynthesis at vents and seeps, from the recycling of nutrients within the deep water food chain. A significant fraction of the total energy budget of the deep ocean is being spent on producing light. This makes bioluminescence not just a biological curiosity, but a major biogeochemical process, one that redirects energy from growth, reproduction, and maintenance into the production of photons that serve ecological functions we have only partially identified. What does the deep ocean do with all that light? Where does the energy go? Most of the photons produced by bioluminescent organisms are absorbed by the surrounding water within meters or tens of meters, their energy converted to heat, an immeasurably small but real contribution to the thermal energy of the deep ocean. But before they are absorbed, those photons carry information. They trigger responses in the eyes and photoreceptors of other organisms. They attract predators and prey. They confuse attackers. They coordinate migrations. They facilitate mating. The photons are the medium through which the ecological business of the deep ocean is conducted. The energy spent on bioluminescence is not wasted. It is the operating cost of the deep ocean's communication network. But what if we are still underestimating what bioluminescence does? What if it is not merely a collection of individual adaptations, lures for predators, camouflage for prey, alarms for victims, but something larger? What if the collective bioluminescence of the deep ocean functions as a system, an ecological organizing principle that shapes the behavior, distribution, and interactions of entire communities? There are accumulating hints that this might be the case. The vertical migration of deep-sea organisms, the daily movement of billions of creatures from the deep to the surface and back again, is the largest animal migration on Earth. It occurs in every ocean, every night, involving biomass measured in billions of tons. and it is accompanied by massive, systematic changes in bioluminescent activity. As organisms rise through the water column at dusk, their bioluminescent displays shift in intensity, color, and pattern. Counter-illumination adjusts as the ambient light changes with depth. Predatory lures are deployed at specific depths where prey density is highest. Alarm signals become more frequent as the ascending organisms enter the twilight zone, where predator density increases. As they descend at dawn, the process reverses. This daily cycle of bioluminescent activity is not random. It is structured, predictable, and linked to the behavior of billions of individual organisms across entire ocean basins. The vertical migration itself is one of the great underappreciated wonders of the natural world. Every evening, as sunset sweeps westward across the planet, a wave of biological ascent follows it. Organisms in the western Pacific rising first, then those in the Indian Ocean, then the Atlantic, then the eastern Pacific, in a rolling wave of upward movement that circles the globe every 24 hours. And with this wave of ascent comes a wave of changing bioluminescence. The deep ocean light regime shifts from its daytime pattern, dominated by counter-illumination and steady, dim glows, to its nighttime pattern, dominated by predatory lures, mating signals, and alarm displays. The entire visual environment of the deep ocean transforms every day, cycling between two distinct photonic landscapes with the regularity of a heartbeat, and then it reverses. As dawn sweeps around the planet, the organisms descend and the bioluminescent landscape shifts back. This is not a local event. It is a global phenomenon. It is the deep ocean breathing, inhaling light upward at dusk, exhaling it downward at dawn, in a cycle that has continued without interruption for as long as the deep ocean has supported life. And the biomass involved in this migration has consequences that extend far beyond the organisms themselves. The billions of tons of organisms that rise and fall each day are transporting organic matter, nutrients, and dissolved gases between the deep ocean and the surface waters. They feed at the surface, where food is abundant, and they defecate, excrete, and die at depth, where their bodies and waste products deliver surface-derived nutrients to the deep. This biological pump, driven largely by the daily vertical migration, is one of the primary mechanisms by which carbon and other elements are transferred from the surface ocean to the deep. It affects global nutrient cycles, carbon sequestration, and potentially even climate regulation. And the organisms that drive this pump, the lanternfish, the krill, the copepods, the squid, are the same organisms whose bioluminescence lights up the deep ocean every night. The pump and the light show are the same phenomenon, driven by the same organisms, serving the same ecological functions. The deep ocean's most important biogeochemical process is also its most spectacular visual display. Some researchers have begun to describe the deep ocean bioluminescent environment as a photonic ecology, an ecosystem in which light itself, rather than chemical energy or physical territory, is the primary organizing force. In a photonic ecology, the fundamental currency is not food or space, it is visibility. The ability to control when you are seen, by whom, at what distance, and in what context determines whether you eat or are eaten, whether you find a mate or die alone, whether you survive or perish. The concept of a photonic ecology has implications that ripple outward through our understanding of deep-sea biology in unexpected ways. If visibility is the primary currency, then the organisms of the deep ocean are not merely producers of light. They are managers of visibility, controlling, with extraordinary precision, the information they broadcast into their environment. Consider the difference between this and how visibility operates on the surface. On land, in sunlight, organisms have limited control over their visibility. A deer in an open field is visible whether it wants to be or not. It can camouflage itself against a specific background, but it cannot turn off the sunlight that illuminates it. In the deep ocean, organisms have complete control. The default state is absolute darkness, total invisibility. Every photon an organism produces is a deliberate act that increases its visibility in a specific direction, at a specific wavelength, for a specific duration. Light production in the deep ocean is not passive exposure, it is active communication, and the organism determines every parameter of the signal. This means that deep-sea organisms are not simply adapted to darkness. They are adapted to a world where visibility is optional, selective, and under individual control. This is a fundamentally different ecological paradigm from anything that exists on the surface. On land, organisms evolve to cope with constant, uncontrollable illumination. In the deep ocean, organisms evolve to manage visibility as a resource, hoarding it when detection would be dangerous, spending it when communication or predation requires it, modulating it in real time as conditions change. The deep ocean is not a dark version of the surface world. It is an entirely different kind of world, organized around entirely different principles, and bioluminescence is the mechanism that makes those principles possible. If this concept is correct, then the deep ocean operates under a set of ecological rules that are fundamentally different from those that govern the surface. On land and in the sunlit ocean, organisms compete for light from an external source, the sun. In the deep ocean, organisms compete using light they produce themselves. They are not passive receivers of illumination. They are active participants in a visual ecology that they themselves create. Every flash, every glow, every pulse is a decision, a communication, with consequences that ripple outward through the surrounding darkness. We do not know how far bioluminescent signals travel in the deep ocean under natural conditions. Laboratory measurements suggest that a bright flash of bioluminescence can be detected by a sensitive eye at distances of several hundred feet in clear, deep ocean water. But the deep ocean is not a uniform medium. Temperature gradients, salinity changes, particle concentrations, and pressure variations all affect the way light travels through water. The effective range of bioluminescent signals may vary enormously depending on local conditions, depth, and the spectral sensitivity of the organism doing the detecting. This variability introduces a dimension of ecological complexity that has barely been explored. If the effective range of bioluminescent signals changes with depth, season, water mass, and local conditions, then the communication environment of the deep ocean is not static. It is dynamic, shifting, and in some ways analogous to the atmospheric conditions that affect radio transmission on the surface. There may be deep ocean equivalents of clear channels, regions or conditions where bioluminescent signals travel exceptionally far, allowing organisms to communicate across distances that would normally be impossible. There may be equivalents of dead zones, areas where suspended particles, temperature gradients, or other factors scatter and absorb light so effectively that bioluminescent signals cannot travel more than a few feet, isolating organisms in pockets of communicative silence. If these conditions exist, they would create a landscape of variable connectivity in the deep ocean, with some regions richly connected by light and others cut off. Organisms would need to navigate this landscape, moving toward regions of good visibility when they need to communicate, and retreating to regions of poor visibility when they need to hide. The geography of light in the deep ocean may be as complex and as ecologically significant as the geography of terrain on the surface. We do not know whether bioluminescent patterns carry species-specific information, the way birdsong identifies species on the surface. Do anglerfish recognize the lure pattern of their own species and avoid it, while being attracted to the lure pattern of an unfamiliar species? Do bioluminescent squid display specific flash codes that identify them to potential mates? Is there a grammar to the light, a set of rules that determines which combinations of flashes carry meaning and which are noise? We do not know whether there are regional differences in bioluminescent behavior, dialects, in effect, that vary between ocean basins, between depth zones, or between isolated populations in different trenches. A species of jellyfish in the Atlantic might produce a fundamentally different light pattern from the same species in the Pacific, the way human populations on different continents speak different languages. If such variation exists, it would suggest that bioluminescent behavior is not entirely hardwired, but is shaped, at least in part, by the local environment, by learning, or by cultural transmission within populations. We do not even know how many bioluminescent species exist on this planet. The most commonly cited estimate puts the number at several thousand known bioluminescent species in the ocean, but this number is acknowledged by researchers as almost certainly a dramatic undercount. Every deep-sea expedition discovers new species. Many are bioluminescent. The actual number of bioluminescent organisms in the ocean could be in the tens of thousands, or the hundreds of thousands, or higher. We simply do not have the data to constrain the estimate. The difficulty of estimating total bioluminescent diversity is compounded by a problem that is unique to this field. Many organisms lose their bioluminescence when they die or when they are brought to the surface. The pressure changes, temperature shifts, and physical trauma of collection can destroy the delicate photophore structures, degrade the luciferin substrates, denature the luciferase enzymes, and kill the symbiotic bacteria that produce light in many species. A trawl net hauled up from 3,000 feet may contain dozens of species that were bioluminescent in their natural habitat, but show no trace of luminescence by the time a researcher examines them on deck. Without video footage from depth, without in-situ observation, there is no way to know which collected specimens were bioluminescent and which were not. The dead, pressure-damaged, light-depleted organisms in a specimen jar are silent witnesses to a capability that was destroyed by the act of collection. This means that our catalogue of bioluminescent species is biased not only by the limitations of our observational tools, which we have already discussed, but also by the limitations of our preservation methods. We are trying to study a phenomenon defined by living light using methods that kill the organisms and extinguish the light. It is as though we were trying to study birdsong by studying dead birds. The anatomical structures are there, the syrinx, the air sacs, the neural pathways, but the song itself is gone, and without hearing it, we cannot know what it sounded like, how complex it was, or what information it carried. The same is true of deep-sea bioluminescence. We can examine photophores under a microscope, analyze the chemistry of extracted luciferins, and measure the spectral output of luminescent reactions in a test tube, but we cannot recreate the flash pattern of a living organism in its natural habitat, at its natural depth, under its natural conditions. The living display, the thing that matters ecologically, is precisely the thing we lose when we collect the organism. What we do know, what the evidence accumulated over the past several decades makes inescapable, is that the deep ocean is not empty, not silent, and not dark. It is filled with life, filled with light, and filled with activity that operates according to principles we have barely begun to decipher. The bioluminescent world beneath 3,000 feet is not a remnant of a simpler, more primitive version of the sunlit world. It is a parallel world, a full, complex, dynamic, and self-organizing system of life that has been running for far longer than any human civilization, in a space far larger than any habitat humans have ever occupied, producing light in quantities and patterns that we are only now beginning to detect. The word parallel is carefully chosen. It does not mean lesser, it does not mean subordinate, it does not mean primitive. The bioluminescent deep ocean is not an earlier version of the surface world, preserved in amber, waiting to be modernized by human understanding. It is a fully realized, fully functional, independently organized world that operates according to its own rules, solves its own problems, and sustains its own inhabitants without any input from or dependence on the surface world above. If every human being disappeared tomorrow, every city, every farm, every ship, every satellite, the bioluminescent deep ocean would not notice. It would continue, unchanged, uninterrupted, unaware. The same flashes would flash. The same migrations would rise and fall. The same alarm signals would spiral through the water. The same ancient chemistry would produce the same ancient light in the same ancient darkness, as it has for longer than any surface ecosystem has existed in its current form. And we have been looking the other way, not deliberately, not maliciously, but through a combination of technological limitation, institutional inertia, and a deep, unexamined assumption that the surface world, our world, the world we can see and touch and walk upon, is the real world, and that the deep ocean is merely an afterthought, a basement beneath the house, a place where a few strange creatures endure, but nothing of real consequence occurs. That assumption is wrong, and the bioluminescent deep ocean is the proof. So what does all of this mean? What does it mean for the way we understand our planet, our species, and our place in the natural world? These are not idle questions. They are the kind of questions that emerge only when the foundations of our understanding are disturbed, when a discovery is large enough to shift the frame through which we see everything else. The first implication is humbling in a way that does not soften with repetition. We do not know our own home. We live on a planet that is mostly ocean, and we have explored a thin, bright fraction of it. We have named and catalogued a small percentage of the species that live in the deep. We have observed an even smaller percentage of their behaviors. We have recorded an even tinier fraction of the bioluminescent displays they produce, and we have understood almost none of the meanings those displays might carry. The bioluminescent deep ocean is not a remote, irrelevant corner of the world. It is the majority of the world. It is the largest habitat, the most chemically active, light-producing system, and quite possibly the most complex visual environment on the planet. And we are strangers to it. This is not a failure of science. Science has achieved extraordinary things in its investigation of the deep ocean, often under conditions of extreme difficulty and with limited resources. The researchers who have descended in submersibles, deployed cameras on the seafloor, analyzed trawl catches, and synthesized observations from decades of expeditions have collectively built a body of knowledge that is impressive by any standard. But that body of knowledge, when measured against the scale of what remains unknown, is thin. It is a sketch, not a portrait, and the sketch has gaps large enough to conceal entire ecosystems, entire categories of behavior, entire dimensions of biological communication. The difficulty of the task cannot be overstated. The deep ocean resists observation in every conceivable way. It is dark, so instruments must bring their own light, which alters the behavior of the organisms being studied. It is cold, so equipment must be engineered to function at near-freezing temperatures for extended periods. It is under immense pressure, so every component, every seal, every window, every cable must withstand forces that would crush ordinary materials. It is far from the surface, so data must be stored or transmitted across miles of water column, and it is vast, so staggeringly, incomprehensibly vast, that even if we had 1,000 submersibles running simultaneously, we could not survey more than a negligible fraction of the total volume in a human lifetime. The economics of deep-sea research compound the difficulty. A single dive by a human-occupied submersible capable of reaching the midnight zone costs tens of thousands of dollars in fuel, crew, ship time, and equipment maintenance. A dedicated deep-sea research cruise, the kind that deploys cameras, collects specimens, and maps a small section of the ocean floor, can cost millions of dollars and produce data that covers an area smaller than a city park. The ratio of cost to coverage is staggering. And because deep-sea research must compete for funding with every other branch of science — medicine, physics, climate science, space exploration — the resources available are never sufficient. Entire ocean basins remain unstudied, not because they lack scientific interests, but because no one has the funding to send a ship there. The bioluminescent world beneath 3,000 feet is not hidden by depth alone. It is hidden by economics, by institutional priorities, and by the simple arithmetic of a research budget that cannot stretch far enough to cover a planet that is mostly ocean. There is also a quieter, more personal dimension to this challenge that deserves acknowledgement. The scientists who study the deep ocean, the marine biologists, the oceanographers, the engineers who design and operate the submersibles and cameras, are working at the boundary of human capability. They are studying a world that actively resists their presence, using tools that are always inadequate and always breaking, in an environment that can kill them if a single seal fails or a single cable snaps. The personal courage required to descend in a submersible to depths where rescue is impossible, where communication with the surface is limited, and where the margin for error is measured in millimeters of steel and glass, is rarely discussed in scientific papers. But it is real, and it is part of the story. The knowledge we have gained about the deep ocean has been won not only with dollars and engineering, but with nerve. The knowledge we have gained about the deep ocean has been won at great expense, great effort, and sometimes great personal risk. It represents a genuine triumph of human curiosity and engineering ingenuity. But it is not enough, and it may never be enough. The deep ocean is so large, so varied, and so resistant to our instruments that a complete survey of its bioluminescent inhabitants and their behaviors may take generations, if it is ever completed at all. Entire species will go extinct before they are discovered, their light patterns never recorded, their ecological roles never understood. Entire communities may shift, migrate, or collapse before a single camera captures their existence. The bioluminescent world that glows right now, at this exact moment beneath every ocean on Earth, is dynamic and changing in ways we cannot track, responding to pressures we cannot identify, and producing signals we cannot receive. The second implication concerns our understanding of life itself and where it might exist beyond this planet. For most of the history of biology, the study of life has focused overwhelmingly on organisms that live in sunlight. The conceptual model that underpins our search for life elsewhere in the solar system and beyond is built on the assumption that life needs a star. We look for habitable zones, the narrow band of distance from a star where liquid water can exist on a planet's surface. We look for atmospheric signatures of oxygen and methane, which on Earth are produced by photosynthetic organisms and their consumers. We design our telescopes and our spectrographs to detect the chemical fingerprints of sun-driven biology. But the deep ocean challenges this assumption at its root. Life does not require sunlight. It requires energy, and energy comes in many forms. Chemosynthesis at hydrothermal vents provides energy from volcanic chemistry. Organic debris raining from above provides energy from the decomposition of dead organisms. And bioluminescence itself, while not an energy source, is evidence of organisms that have built rich, complex ecosystems in the complete absence of solar input. If life can thrive in the permanent darkness beneath our own ocean, it can, in principle, thrive in the permanent darkness beneath the ice shells of other worlds. Jupiter's moon Europa is covered in a thick shell of ice, perhaps 10 to 20 miles deep, beneath which lies a global ocean of liquid water that may contain twice the volume of all the water on Earth. Saturn's moon Enceladus has a subsurface ocean that vents plumes of water vapor and ice particles into space, carrying with them the chemical signatures of hydrothermal activity, silica particles, molecular hydrogen, and complex organic molecules. Both moons are dark, cold, and far from the sun, and both moons contain the basic ingredients that sustain life in our own deep ocean: liquid water, chemical energy, mineral surfaces, and the potential for complex chemistry. The parallel between Earth's deep ocean and these extraterrestrial environments is not speculative. It is grounded in measurable physical conditions. The pressure at the base of Europa's ice shell, where the ocean meets the rocky seafloor, is estimated to be roughly comparable to the pressures found in the deepest parts of Earth's ocean. The temperature of the water is likely near freezing, similar to the abyssal temperatures on Earth. The chemical gradients at the rock-water interface, where heated fluids interact with cold ocean water, may closely resemble the conditions at terrestrial hydrothermal vents. If life exists in these subsurface oceans, and many astrobiologists consider it plausible, it would face the same fundamental challenges as life in Earth's deep ocean: no sunlight, extreme pressure, scarce food, and total darkness. If bioluminescence is a near-universal adaptation to life in darkness under pressure, and the evidence from Earth's oceans suggests that it may well be, then the subsurface oceans of Europa and Enceladus, if they harbour life, may also harbour light. Not starlight, not sunlight, living light. The same cold, efficient, ancient bioluminescent chemistry that glows in the waters beneath our own ships may glow in the waters beneath the ice of a distant moon. We would not see it from space. We might not detect it with any instrument we currently possess. But it might be there, a living glow in the darkness of an alien ocean, on a world circling a planet hundreds of millions of miles from the nearest star. This possibility reframes the search for life beyond Earth in a profound way. We have been looking for life that looks like us, life that lives in sunlight, on surfaces, in atmospheres. But the deep ocean suggests that most of life, by volume, does none of those things. Most of life, on this planet at least, lives in the dark. It does not need an atmosphere. It does not need a surface. It does not need a star. It needs water, chemistry, and energy. And if it is anything like the life in our own deep ocean, it will have found a way to glow. The third implication is perhaps the most personal and the most difficult to articulate. It concerns how we see ourselves as a species, and it asks us to reconsider a story we have been telling for a very long time. Humans have always placed themselves at the center of the narrative. We build our cities on the surface, we farm the land, we navigate by the stars and measure our history in centuries and millennia. We write books about our achievements, our philosophies, our religions, and our technologies. We tell ourselves that we are the planet's most significant inhabitants, that our intelligence sets us apart, that our understanding of the world is comprehensive and growing more complete with each passing year. But the deep ocean suggests a different story. The largest habitat on the planet does not know about us. It does not need us. Its inhabitants have never seen a human face, never heard a human voice, never encountered a human tool except on the rare occasions when a submersible or a remotely operated vehicle intrudes briefly into their world before withdrawing. The bioluminescent ecosystem of the deep ocean was here before long any human civilization. It will be here long after the last one has ended. It operates on timescales that make human history look like a footnote, and it covers a volume of space that makes human territory look like a postage stamp on a football field. There is a particular kind of silence that accompanies this realization. Not the silence of emptiness, but the silence of scale, the recognition that the world is vastly larger, vastly more complex, and vastly more indifferent than any story we have told about it. The bioluminescent deep ocean does not care about our theories. It does not pause for our instruments. It does not adjust its rhythms to accommodate our research schedules. It simply continues, flashing, glowing, pulsing, signaling, in a darkness that has no interest in being illuminated by our understanding. And perhaps that is the most important thing to take from all of this. Not any single fact about luciferins or photophores or counter-illumination or quorum sensing. Not the specific number of species discovered, or the particular depth at which a camera recorded a squid attacking a lure, but the simple, overwhelming, and slightly terrifying recognition that we are not the main event. We are a recent, surface-dwelling species, living on a thin, dry crust above a world of darkness and living light that we are only now beginning to notice. The deep ocean was here before us, and it will be here after us, and its light will continue to burn in the darkness long after our own lights have gone out. Somewhere right now at this very moment, in the waters of the Atlantic or the Pacific or the Indian Ocean, at a depth where the sun has never shone and never will, something is glowing. It is not a lamp, it is not a fire, it is not a reflection or a trick of refraction. It is a living organism, producing its own light in the darkness, under pressure so immense that the water around it is denser than anything on the surface of the earth. It is sending a message. Perhaps it is a warning, a signal to a predator that says, "Do not come closer, I am not worth the effort." Perhaps it is a lure, a false promise of food, dangling in the void, waiting for something hungry to approach. Perhaps it is a love letter written in photons, cast into the blind water in the hope that something, somewhere in the surrounding darkness, will see it and respond. Perhaps it is a cry for help, an alarm thrown outward in all directions, calling for a rescuer that may or may not exist. Or perhaps it is something else entirely, something that does not fit any category we have imagined, serving a purpose that no human mind has yet conceived. That light has been burning for a very long time, long before any human took a breath, long before any city was built on any continent, long before any ship set sail across the surface of the water above, long before any scientist asked what might live in the deep, long before the first cable was dropped overboard to measure the depth, long before the first net was dragged through the midnight zone, the bioluminescent world was already ancient. It was already vast, it was already complex, and it was already conducting its business, its predation and its courtship, its deception and its defense, its birth and its death, in a language of light that no surface-dwelling creature had ever witnessed. The bioluminescent world beneath 3,000 feet is not new. It is one of the oldest continuous biological phenomena on this planet. The chemical reaction that produces bioluminescence, the oxidation of luciferin by luciferase in the presence of oxygen, is among the most ancient biochemical processes still operating in living organisms. The molecules are old, the enzymes are old, the organisms that carry them have been producing light in the deep for longer than many mountain ranges have existed, for longer than most surface ecosystems have maintained their current form. The deep ocean, with its stable temperatures, its stable pressures, and its unchanging darkness, has provided a consistent environment in which bioluminescence could persist, diversify, and spread across thousands of lineages without interruption. What we see today when we lower a camera into the midnight zone is not a new phenomenon. It is the latest chapter in a story that has been unfolding continuously in the dark for an almost inconceivable span of time. The stability of the deep ocean is itself a remarkable fact. While the surface of the planet has undergone dramatic transformations, continents drifting, ice ages advancing and retreating, forests rising and burning, entire categories of surface life appearing and disappearing, the deep ocean has remained essentially unchanged. The temperature at 4,000 feet in the mid-Atlantic is virtually the same today as it was millions of years ago. The pressure is the same. The darkness is the same. The chemical composition of the deep water changes slowly, over timescales that make human history vanish into insignificance. In this unchanging world, bioluminescence has had the time and the stability to develop a complexity that surface phenomena, subject to constant disruption, have rarely achieved. The deep ocean is a laboratory that has been running the same experiment, life in darkness, illuminated by its own chemistry, for longer than any other experiment on this planet. And the results of that experiment are glowing beneath us right now, at this very moment, in waters that most of humanity will never see. And almost no one has seen it. This is the paradox that defines our relationship with the deep ocean. We live on a planet where the dominant form of biological light is not the light we know, where the majority of visible photons, in terms of the sheer volume of space in which they are produced, are generated not by the sun, but by the chemistry of living cells, where the largest and most widespread visual communication system operates not in the air, not on land, not in the shallow waters where humans swim and fish and play, but in the cold, pressurized, sunless depths that most people will never see and never think about. We live on a planet of living light, and we have spent most of our history looking the other way. But the ocean knows. The ocean has always known. Beneath the surface that sailors cross, beneath the waves that crash on beaches, beneath the shallows where coral grows and fish school in the familiar, sun-dappled water, there is another world, a world that does not need the sun, a world that makes its own light on its own terms, for its own reasons, a world where communication happens in flashes and pulses, where predation is conducted by glow, where survival depends on the ability to control photons in a darkness so complete that the concept of day and night has no meaning. Every night, as the sun sets above the ocean surface, something extraordinary happens. Billions of organisms, fish, squid, shrimp, jellyfish, worms, larvaceans, copepods, siphonophores, and countless others, begin their ascent from the deep. They rise through the water column, from the midnight zone toward the twilight zone, and beyond, in the largest daily migration of living things on the planet. This migration dwarfs the wildebeest herds of the Serengeti, the caribou migrations of the Arctic, the flocking of starlings in European skies. It involves more individuals, more biomass, and more vertical distance than any other animal movement on Earth. And as they rise, these creatures carry their light with them. The deep ocean does not stay in the deep. It reaches upward every single night, carrying its bioluminescence into shallower waters, bringing its signals closer to the surface, narrowing the gap between its world and ours. And then, as dawn approaches, it reverses. The organisms descend, pulling their light back down into the darkness, retreating to a depth where no human eye can follow, where no surface instrument can observe them without disturbing them, where the light they carry disappears once again beneath the weight of the water. This has been happening every day and every night in every ocean for as long as the deep ocean has supported life. It is happening right now, at this very moment, beneath the cargo ships that cross the Pacific, beneath the fishing boats that rest at anchor in the Atlantic, beneath the swimmer who floats on the surface of the Mediterranean and looks up at the stars. The stars are beautiful and they are very far away. But the lights below are closer, they are alive, they are ancient, and they are telling stories that no one on the surface has ever heard, in a language that no human mind has ever decoded, about a world that no human eye has ever fully seen. There is a world on this planet where the sun has never reached, and in that world, something is glowing. Something has always been glowing, and it will continue to glow long after the last question has been asked and the last answer has been given. The light does not wait for us. It does not need our attention, our instruments, or our understanding. It simply persists, ancient, patient, and continuous, in the largest darkness on Earth. The ocean is not silent. It is not empty. It is not dark. It is the most luminous place on this planet, and we have only just begun to see it. The only question that remains is whether we will find the courage, the patience, and the humility to look down into the darkness and learn what the light has been saying all along. We have spent centuries looking outward, at the stars, at the planets, at the distant galaxies that recede into a cosmos too large for comprehension. We have pointed our telescopes at the sky and asked the universe to reveal its secrets. And the universe has obliged in its fashion, offering us galaxies and nebulae and the faint afterglow of creation itself. But we have neglected to look down. We have neglected the world beneath our own feet, a world that is closer, more accessible, more alive, and more strange than anything we have found in space. The bioluminescent deep ocean is not waiting for us at the end of a long journey through the void. It is here, it is now, it is directly below the surface of the water that laps at the shores of every continent on Earth. And it has been glowing, patiently, continuously, without interruption and without acknowledgement, for longer than any human culture has existed. There is a kind of grace in that patience. The deep ocean does not demand our attention. It does not perform for our cameras. It does not adjust its rhythms to suit our schedules or our funding cycles. It simply continues, and the light continues with it, ancient, patient, and indifferent to our awareness of it. One day, perhaps, we will learn to see it as it truly is. Until then, it will keep glowing. It has always been glowing, and the darkness, which is not empty, will carry the light forward into whatever comes next, as it always has, in the deepest and most luminous silence on Earth.